Yeah, it has come.
Yeah, so they will be joining soon. We can wait for 1 minute or so. So, okay, good afternoon, everyone. Let's get started and welcome to second session of this national conference, which is host-pathogen interaction, present and future perspective. We had a pretty good first session where Professor Dysregulated and Arun Sharma, along with Balasubramani GL, presented their research findings. Now we are fortunate enough to have Dr. Anirban Banerjee with us, and his talk will be followed by talk by Professor Dipankar Nandi from IISc Bangalore, followed by Dr. Sandhya Marathe from Pilani and Dr. Shabir Ahmed from THSTI. As far as Professor Anirban Banerjee is concerned, he did his bachelor's in science from Calcutta University. And then to get the flavor of North India, he moved to Punjabi University, Patiala for doing his master's. And then I think so stayed there a little bit longer to pursue his PhD from NIPER. And then went on to do his two postdocs. One was from University of South Dakota, followed by another stint in San Diego State University. And then I think so for a year or so, he was in DuPont Knowledge Center at Hyderabad, before joining IIT Bombay as assistant professor in 2012. And right now he's associate professor in Department of Biosciences and Bioengineering. And the primary research focus of his lab is bacterial pathogenesis and host pathogen interactions, particularly those pertaining to Streptococcus pneumoniae and Group B streptococcal infections. He is a recipient of Early Research Achiever Award and Best Teacher Award from IIT Bombay. And apart from that, I think so, he is also publishing in very good reputed journals from there. And hopefully, we are fortunate enough to have him on board. And whatever the experience he will share with us, I think so, all of our community will also be benefited along with the past participants of this conference. So, Professor Anirban Banerjee, please.
Well, thank you, Rohan, for the nice introduction. And thank you all of the organizers, for arranging this two-day conference, national conference on host-pathogen interactions, which is very rare in India, I suppose. Generally, we have conferences on cancers and all other, but host-pathogen interactions is very rare. And I hope all of you are well in this, amid this pandemic, which is unprecedented. We haven't seen. Of course, in our life, definitely we haven't seen, and our predecessors' life also, they haven't probably seen this thing happen. So I will talk to you something about Streptococcus pneumoniae, a story which has been evolving in our lab for the past few years. And hopefully you will be able to appreciate at the end of the talk that whatever I'm trying to convey over here. The title is extremely, I think it's a little confusing in terms of like what makes enemies if we can be friends. So I hope by the end of the talk, you will be able to understand what do I exactly try to mean by this particular title. So I'll share my screen and we will start with the presentation. Is it visible and am I audible?
Yes, Anirban. Yeah, it's fine.
Okay, so as I said, the title is Why Make Enemies and If We Can Be Friends. So myself is Anirban Banerjee and I am from Department of Biosciences and Bioengineering at IIT Bombay. So whenever we talk about bacteria and a host, that means a host and a pathogen, generally the thing that comes to our mind is that there is an arms race in infectious disease biology. That means the bacteria is trying to produce different virulence factors such as different toxins, pore-forming toxins, hydrogen peroxide, other soluble factors which are cleaving the host secreted antibodies and all that, as well as the host is trying to recapture the bacteria, trying to kill them by having influx of phagocytic cells such as macrophages or neutrophils, or secreting a lot of cytokines and chemokines and other proteases by which they are trying to degrade the bacteria. And as we have seen during the entire course of evolution, both the bug as well as the host are constantly evolving and trying to give them a trump card against each other. So the host factors, as I said, macrophages, neutrophils, antimicrobial peptides, whereas the bacterial factors could be toxins, bacteriocins, and all that. And in between all this, if the host is able to kill off the bacteria, there is a lot of inflammation. And that is given over here, that inflammation is there. And after the infection is cleared, then comes a repair and a resolution phase of that particular tissue, wherever the inflammation was taking place. But in case the inflammation goes dysregulated and it lasts for long, it actually does a lot of harm to the host itself. And sometimes it causes mortality. That brings us to this particular conundrum, I would say. What do we call as a good bacteria versus a bad bacteria? So if you can look at the middle graph where it is plotted pathogen load versus the health of an individual. And if the pathogen load is low, both the host as well as the bacteria is happy. They are not disturbing each other. But in case the bacteria is a bad bug or so-called pathogen, we can say, and if the pathogenic load is higher, then the health of the individual goes really bad. And it's probably leads to sick person or maybe lead to mortality and death person. Whereas if the pathogen is not a pathogen, typically what we call as the gut microbiome or gut microbiota, that significant number of that, even though it is present, it does not cause any significant harm to the host health. And in that case, the host can be a very happy, healthy individual. So there is this conundrum, what the bacteria is willing to do, whether it wants to become a good pathogen for the host or whether it wants to become a bad pathogen. Now, from a bacterial perspective, it doesn't make sense for the to kill the host, because if at all it does, then it leads to the loss of reservoir. And this is particularly important for an obligate symbiont such as Streptococcus pneumoniae. And that is our bug of interest. So Streptococcus pneumoniae is an asymptomatic resident of the human nasopharynx, and that is the only environmental reservoir it has. Of course, it is the only human, it is primarily a human pathogen, and there are 97 different serotypes of this particular thing is known. And if at all you can see this, this is the nasopharyngeal immunohistochemistry, and you can see that Streptococcus pneumoniae nicely lining on the nasopharyngeal epithelium of the human nasopharynx. So under certain conditions, these biofilms disperse and the transition happens from the biofilm in an air droplet-like fashion, and it travels from one individual to another individual. And that's how the transmission happens. Now, understandably, what I said earlier, if pneumonia leads to the loss of the particular individual or the individual dies because of pneumococcal infection, that essentially means that is a dead end for strep pneumonia, because it cannot transmit from one individual to another individual through any other body fluids. The only means of transmission from one individual to another individual is through air droplets. That means whenever we cough or sneeze, then only it transmits. So it makes sense for the bug to keep the host alive. And one of the major virulence factors, if at all we can call it a virulence factor, is a protein toxin secreted by this bacteria, which is name is pneumolysin. Now, as shown in this particular figure, Pneumolysin is actually secreted by pneumonia in a monomeric fashion, and this pneumolysin actually identifies the host cell surface cholesterol and aggregates on the host cell surface to form an oligomer. Once an oligomers are formed, 32 monomer comes together and forms an oligomer, then there is a conformational change that happens in pneumolysin, and it makes a plunge into the lipid bilayer of the host cells, and thereby it forms these pores. And because of this pore, the host cell is lysed, the intracellular contents come out, and actually that leads to massive inflammation. So this inflammation has two roles. Number one, this inflammation is understandably responsible for the mortality and the loss of occupiable habitat, as I have initially explained. But if this inflammation is also very much important for the transmission, because if at all you do not follow this inflammation, then you are not going to sneeze and cough, and if at all you do not sneeze or cough, you are not going to transmit the bacteria from one individual to another individual. So for transmission to the new host, this very regulated inflammation is absolutely critical. Now comes the very important question, what if at all I take away this transmission cost? So essentially this is a cost to the bacteria, and if at all I take away that cost from the bacteria, whether that would lead to evolution of Streptococcus pneumoniae or this particular virulence factor. That was the cause, that was the question that we asked. And when we started looking at different Streptococcus pneumoniae clinical isolates, we found out that there are approximately 20 different alleles of these very important virulence factors of pneumolysin. So now, if this particular virulence factor is so much important to the bowel, why it would have so many variants out there in all clinical isolates of Streptococcus pneumoniae? And what we found out is one of the major clinical isolates, which is globally more prevalent, that is named as ST306, which also expresses this pneumolysin. But this pneumolysin does not lyse the red blood cell. That means the hemolysis percentages is absolutely zero or negligible compared to other clinical isolates which are expressing a hemolytic pneumolysin. But then this particular clinical isolates, which is ST306, was actually able to transmit from one individual to another individual by transmission via coughs or sneezes, and they generally do not cause lethal infections. So they do cause infections, but they are sublethal, but they are very important for transmission from one individual to another individual. And the only difference between this non-hemolytic pneumolysin, which is given by this allele 5 versus allele 1 is over here, is they are different by 3 amino acid substitution and 2 deletions. So you must appreciate the fact that the only 5 changes in a 471 amino acid long sequence is sufficient to completely abolish the hemolytic activity of this protein toxin. So we wanted to first ask the question whether this particular toxin, which is not able to cause hemolysis, whether it at all forms pores on the host cellular membrane. So we used a very elegant technique where we encapsulated calcine, which is a fluorescein dye, in a liposome in a quenching concentration. So these liposomes will not fluoresce unless and until there is a pore in this, and pneumolysin mediated pore can allow the calcine to be released from this particular liposomes, and then only they can fluoresce. And you can see from this graph, all cases the PLYH, that means the hemolytic pneumolysin, which is coming from the allele 1, they all create calcine release. So quick release of calcine, and that's why fluorescence intensity is increased. Whereas non-hemolytic pneumolysin, which is given by this PLYNH, NH stands for non-hemolytic, H stands for hemolytic, there is no release of calcine. And you can see only release of calcine when we put Triton X100, which is a positive control. But now we ask the question whether it is okay, that essentially means it is this particular non-hemolytic pneumolysin is not able to form pores on this liposomes, but whether they are able to bind or not. So we have done some Western blotting, and you can see that both the binding of hemolytic and the non-hemolytic toxin are competitively inhibited by presence of cholesterol, because both of this pneumolysin is known to bind to the cholesterol on the host cell surface and then oligomerize. And you can see from this transmission electron micrographs that the PLYH as well as PLYNH both form nice oligomeric structures on the host cell surface, albeit there is some
difference in the structures of these two, it looks like that the PLY-NH are actually standing on the membrane and an elevated form projected outside the membrane. At least that is what it looks like from these two-dimensional transmission electron micrographs. We then did the animal experiments straight away. And the first thing we did is the study of the survival of the animals whenever we infected with the hemolytic version or a non-hemolytic version. And in case the hemolytic version where we have allelic exchange, the hemolytic pneumolysin with the non-hemolytic pneumolysin. And to our surprise, we saw that the non-hemolytic version, no mice actually died. Very few, only one or two. And wherever we did the allelic exchange, few mice only survived and few mice were dead. It is in between the phenotypes. So that essentially showed there are other factors also involved in a disease progression. Now, when we did the lung bacterial count from these infected mice at different time course, and you will see over there that initially the hemolytic pneumolysin was extremely high in count in the lung, and so the lung counts were clinically going down. And after 96 to 168 hours, when there was no hemolytic pneumolysin containing mouse were alive, and only non-hemolytic pneumolysin mice containing were alive, you could see that you could still recover substantial amount of the bacteria from the lungs of these mice. So this was very perplexing to the fact, because generally the mice is alive over here in the survival curve because of either one reason, that the mice is able to clear of the infection. But this lung counts gave us an idea that it is the bacteria is actually surviving in the lungs of the mouse, though they are not being cleared from the mice lungs. So what we did, we actually went on to do the inflammatory response from the lungs of these mice, and we could see that the lungs of these particular PLY-H, that means the hemolytic pneumolysin containing mice, showed significantly higher amount of inflammation compared to the non-hemolytic infected mice. And you can see from this immunohistochemistry over here of the lungs of the mice that the PLY-H containing mice, the alveolars is shrunk, and there is lots of infiltration of the neutrophils and the other leukocytes compared to non-hemolytic pneumolysin or the clinical strain ST006, where the alveolus you can clearly see the epithelium of the alveolar and the air sacs you can clearly view. So the mice was healthy, it was surviving for a long period of time when they were infected with PLY-NH or ST306, but there was substantial number of bacteria that is being present in the lungs of the mice. So at that point of time, we were very curious, then how come this, where are these bacteria in the lungs of these particular animals? So we took the lungs and we sectioned them, these particular lungs, and saw them under transmission electron microscope, only to find out, this is typically known as serial block phase imaging. So, and we can see that the bacteria is present inside the lung epithelial cells. These are the lung sections following 168 hours of animal survival. So these animals were absolutely perfect. They were jumping up and down. And you can take these animals, section their lungs, and you can see this bacteria present inside the lung epithelial cells in the lungs of these mice. We went on to do a transmission electron microscope of these particular lungs, and you can see very clearly bacteria being present inside the lung epithelial cells over here in the down picture. So now we did an assay that approximately 72 to 96 hours after infection, we did sacrifice the animals and did a lavage of this bronchoalveolar lavage and calculated the tissue CFU versus the lavage CFU, which will give an idea about whether the bacteria is indeed present inside the lung epithelial cells, whether the bacteria was just floating outside the epithelial cells in the lung. And you can see over here that both in case of PLY-NH and ST306, significantly higher amount of tissue CFU or lavage CFU was recovered. That essentially means that the bacteria whichever was present at higher number in the lung tissue at 168 hours was basically due to the fact that they were present inside the cells. And it makes sense because once they're present inside the cells, they can survive because on the face of the threat of the inflammatory cells being present in that particular environment. And quite a few of the antibiotics, even if you take, they will not penetrate the host cells. So this is a perfect hiding place for the bacteria for a long period of time. A lot of intracellular pathogens actually do it. But we were extremely surprised to see Streptococcus pneumonia behave in this particular fashion because this is generally known as an extracellular pathogen. So how come this PLY-NH is enabling the bacteria, that means Streptomyces, to go inside the host cells. So generally we know that the host cell undergoes different pathways, endocytic pathways, in order to take up the foreign cargo, which involves phagocytosis, bacteriopinocytosis, clathrin-coated vesicles, clavulin mediated pathway, and all that. So we thought that there might be two things that might happen over here. Either the PLY-NH, having PLY-NH is actually conferring some advantage to the bacteria so that it can get inside the host cells, or PLY-H was negatively impacting the endocytosis of the host cell. So we tried in lung epithelial cells, and all these experiments henceforth were done with lung epithelial cell line A549. These are in vitro experiments, and we have done some invasion efficiency, and you can see the invasion efficiency of the PLY-NH containing bacteria was significantly higher compared to the PLY-H. Now we treated the host cells with either purified PLY-H and NH and infected with the opposite cells. So if at all we are treating the cells with PLY-H, then we are infecting them with the PLY-NH, like that. And so we saw that whenever we treated the cells with PLY-H and then infected with PLY-NH, the advantage that we had in case of PLY-NH invasion, that was lost. So essentially, PLY-H is detrimentally affecting the endocytosis process of the host cells. And since we know that the PLY-H segregates to the lipid raft and possibly binds to cholesterol, that's why we check whether the advantage that we have with PLY-NH being present, whether we can nullify that by blocking the caveolae or lipid raft mediated endocytosis. So whenever we treated the cells with beta-cyclodextrin, you can see that advantage of the PLY-NH had over the PLY-NH in terms of endocytosis, that was completely nullified. We then studied whether both PLY-H as well as PLY-NH, whether they can both segregate to the lipid raft, and as a positive control, we took cholera-toxin B, which is known to segregate to the lipid rafts, and as a negative control, we took transferrin, which is generally getting internalized to clathrin-mediated endocytosis. So you can see over there that PLY-H as well as PLY-NH both segregates to the lipid rafts. So at this point of time, we hypothesize maybe PLY-H as well as PLY-NH, both are binding to the lipid raft, but PLY-H, because of its pore-forming ability, may be disrupting the lipid rafts, and that's why the endocytosis is low, whereas PLY-NH, though it binds to the lipid rafts, since it is not forming pores, it is not disrupting the lipid rafts, and that's why we could see increased amount of endocytosis in case of the bacteria, which is harboring the PLY-NH. So in order to study that, what we did, we coated latex beads, inert latex beads with cholera-toxin B, and studied its internalization following treatment with PLY-H and NH. And M-beta-CD served as a positive control. So whenever we treated M-beta-CD, there were no beads which were internalized into the host cells, because both the beads got stained by both the antibodies, which was green as well as red. So this is differential staining. But whenever you will see that it is treated with PLY-NH, we could see red beads. That means those beads which have gone inside. Whereas PLY-NH, no beads were red. That means none of the beads actually went inside. So our hypothesis was correct that both the toxin, both the forms of the toxin, both PLY-H as well as PLY-NH was segregating to the lipid rafts. But PLY-H, because of its pore-forming activity, was creating punching holes onto these lipid rafts, and that's why it was stopping endocytosis or inhibiting endocytosis of the bacteria. Now, once it is endocytosed, I have shown in the in vivo studies the bacteria was staying inside the lung epithelial cells for 168 hours, which is like for 7 days. So once it gone inside the host cell, it has still to survive in the hostile environment of the host intracellular defense mechanisms. And that generally involves the phagocytic mechanism, that means fusion with the lysosomes, and autophagy. And autophagy in this particular case kicks in because following internalization, the bacteria would punch holes on these endosomal membranes because of the expression of this pore-forming toxin, which is pneumolysin. So if the bacteria punches hole on this endosomal membrane, this endosomal membrane is ruptured, and that exposes the luminal glycan molecules to the host cytosol. And the host can bind to this a particular protein called galactin-8, which recognizes this cytosolic or the luminal glycal molecules. The galactin-8 is a danger receptor or danger signal. It in turn binds to autophagy receptor NDP52, which in turn binds to autophagic molecule, which is LC3. And that's how the host again can sequester the bacteria in an autophagosome and finally fuses with the lysosome in order to kill it. So essentially this means if PLYH, which can form such pores, will co-localize with galactin-8 as well as LC3, whereas PLYNH, which does not form some endosomal damage, will not co-localize with GAL8 and LC3. And that's what essentially we see. Whenever we will use PLYH, it will co-localize with GAL8 and LAMP1, which is a marker of lysosomes, whereas PLYNH will neither co-localize with GAL8 nor with LC3 nor with LAMP1. On the other hand, once this galactin-8 is not done, then also there is a secondary mechanism of the host cell ubiquitinates the bacteria. So PLYH, again, because it forms damage in the host cell endosome, it can co-localize with ubiquitin, which in turn co-localizes with or associates with LC3 and LAMP1, whereas PLYNH does not. So pore formation by PLY is essential for damage and subsequent association with cellular danger signals. But whether they actually lead to bacterial killing, that's what is important. So we actually did time scale analysis of association of bacteria with GAL8 or ubiquitin, and you can see PLYNH containing bacteria will minimally associated with GAL8 and ubiquitin. And in case of PLYH, most of the GAL8 or ubiquitin positive bacteria are associated with LC3. So that essentially means whether the damage was actually diverting the bacteria towards to be recognized by the host cell danger receptors such as the GAL8 or ubiquitin, which in turn was associating with LC3, and autophagosomal degradation of the bacteria was happening. So this means that the PLYNH containing bacteria, which is not able to induce such damage, probably would live longer, and our intracellular survival assays exactly shows that in the lung epithelial cells. You please concentrate on these gray bars and versus these white bars, and you can see at extended period of time, the PLYNH containing bacteria was able to survive at much higher capacity compared to the PLYH containing bacteria. Sorry, the gray bar should be PLYH. There is a mistake over here. Now we also did I said that the difference between PLYNH and H was only 3 substitutions and 2 deletions. And out of these 3 substitutions and 2 deletions, only 2 amino acids were majorly important. We have found out by structural analysis. By that, these 2 particular substitutions were majorly important in the pore forming activity. So whenever we revert back those 2 substitutions towards the allele 1, we could see that the bacteria could again form pores, and those pores were responsible for lower survival of the bacteria inside the host cell. So loss of pore forming ability confers longer intracellular persistence inside the host cell. And our earlier studies have shown that there is heterogeneity in the pneumolysin population. So if at all you grow Streptococcus pneumonia wild type strain, you will see there are majority of the bacteria are actually not producing pneumolysin, and very few are producing pneumolysin. And our animal studies has shown that these particular low pneumolysin producers are actually more able to cause disease. So what we thought that whether these if at all we keep on recycling this bacteria towards this host intracellular defense mechanisms, whether that would actually lead rise to this low hemolytic or non-hemolytic phenotype. And to do that, what we did, we continually passed through the cells. We allowed the pneumococcus to get inside the host cells, killed the outside bacteria, harvest the intracellular bacteria, and again reinfected them. So basically by doing this, we are trying to enrich the population, which is the intracellular bacteria. This is typically known as in vitro evolution, and we did that for 20 passages, and following 20 passages, we isolated 23 clones, and out of those 23 clones, 10 clones showed considerably lower amount of pneumolysin expression or hemolytic activity compared to the wild type strains. But we did not find a single mutation in the pneumolysin gene. So then we went to do the similar experiment in an in vivo setup. This is typically known as in vivo evolution. So what we did is that we took the wild type pneumolysin, wild type bacteria, which is 100% hemolytic, forms spores, and we divided into 10 different tubes. With each different tube, we infected one animal, isolated their lungs after 48 hours, plated the bacteria on a plate, and again infected the animals. So like that, we infected 10 different groups, 10 different animals for 20 passages. That means 20 animals it went through in each group. And after 20 animals, after 20 passages, we isolated 10 different lineages. And you can see then in case of 7 out of 10 different lineages showed extremely low amount of pneumolysin expression. This is done by pneumolysin ELISAs. And again, very importantly, we could not find a single mutation in the pneumolysin gene. So there might be a possibility that the pneumolysin expression was low because there might be mutations in the pneumolysin regulators, not a single one of such regulators still known till date. We took these evolved strains. This comes the most important thing. We took those evolved strains out of those 10 different strains, 10 different lineages. We took 3 different lineages, lineage number 2, lineage number 1, 2, and 10. So 10, 1, and 2, which showed minimal amount of pneumolysin expression. We took all those 3 strains and actually performed a survival assay. And to our surprise, though all those 3 strains showed improved survival of the animals compared to the wild type strain. Not only that, we counted the bacterial burden in the lungs of these mice, and we found that in all these cases, the wild type animal did not survive beyond 48 hours, but the mutant animals, this particular evolved strain, could survive for longer period of time. And up until 48 hours also, we could see a much large number of bacterial burden in the lungs of these mice, whereas these animals succumbed early. Immunohistochemistry suggested that the lungs of these particular animals, of the wild type infected animals, where there were lots of infiltration of the leukocytes and macrophages and neutrophils, whereas this evolved strain showed minimal damage in the lung. That means it's the inflammation which was doing the damage. And of course, inflammatory cytokines and the leukocytes, if at all we count in the lungs of the mice to facts, we could see higher amount of inflammatory cytokines and leukocytes being present of the mice infected lungs, or the mice that were infected with PLYH. So this comes to our very end, and we could think of an evolutionary trajectory that the pneumococcus is undergoing. It probably at some ages, quite a few thousand years back, it was possibly an opportunistic accidental pathogen. And now, because of mutations probably in PLY regulators, this is an assumption, we are assuming that there is reduced amount of PLY expression, which are possibly showing phenotypes such as persister cells. And further mutation would lead to, in the gene of the pneumolysin, and would lead to reduced PLY activity. And these are the clinical isolates which are actually present in the nature. Additional mutations in the PLY gene would lead to non-hemolytic phenotypes such as the ST306, which is the most globally circulated strain of pneumococcus, which causes sublethal to non-lethal infection. But as I said, it is most globally present strain. That essentially means its transmission ability, like SARS-CoV-2, is possibly the highest among all the clinical isolates of pneumococcus. This evolutionary trajectory is possibly leading it towards commensalism. So it is trying to get into a new niche in the host, which is the lungs of the host. And why it will make so? Is there any relevance with other examples in the nature? So there is a similar bacteria from the similar genus Streptococcus, which is known as Streptococcus mitis. Now Streptococcus mitis is a commensal in our mouth, and it is known that several thousand years ago, Streptococcus mitis, which is over here, is Streptococcus mitis actually evolved from a Streptococcus pneumoniae like ancestors. Now, mitis also actually expresses a similar toxin such as metylicin, but that metylicin does not form pores. And that is the reason probably that mitis can actually stay with human host in a collegial manner. And it probably happened because at quite a few thousand years ago, the human population size was extremely small and mitis could not afford to kill whatever human host it had. So it had only 2 options, either to reduce its virulence by lowering down its pore forming or abolishing its pore forming activity so that it can present in as many number of hosts as possible. And that is what probably it did. And loss of colonization would lead to induced immunity. So what we are seeing in the present day, probably because of the advent of vaccinations or the antibiotics that we are using against Streptococcus pneumoniae, we are watching that there is a resurgence in pneumococcal serotypes such as serotype 1, which does not have a nasal colonization phase. All ST306 does not colonize in the nasal phase, and we do not know where it colonizes, but generally it is known that it acts as a primary pathogen. But it causes, as I said, it causes mild infection, non-lethal or sublethal. But definitely preserves host numbers. And this is the serotype where we see maximum number of pneumolysin variants with low or no hemolytic activity at all. But understandably, as our data showed, this showed improved intracellular life compared to the existing other clinical isolates of Streptococcus pneumoniae, which has a hemolytic version of the pneumolysin. So now, whether this all is leading to a serotype 1 of pneumolysins having a non-hemolytic version, acquiring a new lifestyle in an alternate host, alternate niche of the host, because once you are doing vaccinations, you are actually taking the pneumococcus away from its home. So it has only two possibilities. Either it can go extinct or it should fight for a new niche in the host. And our study shows instead of becoming extinct, it is changing expression or lowering down the pore-forming activity of its most important virulence factors and probably searching for a new host, a new niche in the host. And that new niche is actually our lungs. So even though there are tons of bacteria that are present in those lungs of those animals, the animals are not dying, the animals are not feeling sick, they're happily jumping around. What would that lead to in future? The timescales are not known, but that might lead to longer persistent and a latent infection kind of a thing. Of course, sublethal. It is typical Mycobacterium tuberculosis-like phenotype. So are we watching a tuberculosis-like evolution of pneumococcus over here? That's debatable. I cannot comment on that because we have not seen the time scales in the evolution, but that would certainly lead to antibiotic tolerance or resistance like phenotype, because if at all it stays inside the host cells for longer period of time, it is shown already that bacteria which has longer persistence inside the host cell leads to antibiotic tolerance or resistance. And that adaptive evolution can lead to pneumococcus in the future time course become an intracellular pathogen rather than an extracellular pathogen. And with that, I will end. This is my group. I would like to thank all of them for contributing towards this study, particularly Anjali Anil, who is in the center, who has contributed. This is primarily her study. And I'd like to thank my collaborators, Dr. Dan Neil from University of Liverpool, Dr. Kwang Sik Kim from Johns Hopkins University, Professor Prasenjit Bhaumik, and Professor Devshika Chakraborty from IISc, and all the funding agencies for supporting our studies. And thank you all for your kind attention. I would like to take any questions if at all you have. Thank you.
Thanks, Dr. Anirban, for such a nice and wonderful talk. And I request you to please stop sharing your slides if you can. And first of all, I request all the dignitaries who are joining through Zoom meeting, if you have any questions, please unmute yourself and you can ask from Professor Banerjee if you have any questions.
Yeah, I have a question, Dr. Banerjee.
Yeah.
See, when you're looking these evolution happening, because I was thinking in that direction, and by the moment your slide came where they are going for the commensalism. So the bacteria are preferring to go for the commensalism. So these clinical isolate what you have got, how much loss of virulence they have? Are they like significant variation in term of the infectivity and the symptom in the human being, or not?
So if at all I take it, thanks for that. Very nice question, I would say. The infectivity of this ST306 strain, which is a clinical isolate, is very, very low. So we have shown in the animal models also that almost near to negligible death of the mice happens. It causes infections, but those infections are absolutely non-lethal in nature. But you will suffer infection because that is essential for the bacteria to transmit. Because if there is no infection, then you will not cough or sneeze, and that coughing and sneezing is absolutely essential for it to transmit from one individual to another individual. But none of those infections are lethal. So it is sublethal, clinically sublethal.
Okay, another question. When you're saying about this low expression of these pore forming this pneumycin, huh? So what is the regulatory part over there? Like who is regulating?
That's a great question. So just to tell you that, Griffith used this bacteria to prove
right.
DNA as a genetic bacteria. And it's been more than 100 years now. 1920, he did this experiment, means more than 100 years. And we do not know this is the virulence factor of this particular bacteria, but we do not know anything about the regulation of this particular toxin. In fact, we are the first one a couple of years back to show that in a population of Streptococcus pneumonia, even if you culture it in vitro, you would see extreme heterogeneity in terms of phenotypic expression of this bacteria within a population.
Okay. Okay.
And majority of the bacteria was actually not producing pneumolysin. Very few were producing, and those who were producing was exceptionally in high quantity. But to answer your question, we do not know anything about its regulation. We believe that these lower amount of pneumolysin producers are possibly because there is a mutations in the regulatory elements, but we don't have any clues for that. We certainly know that there is no mutations in the gene of that particular toxin.
Okay. Okay. Thank you. Thank you, Dr. Nice talk. Yeah.
Dr. Banerjee, is there any role of the host factors in doing this, in having this transition from produced?
Go ahead, Ron.
Yeah, from a strain, which is producing, and from a strain which is not producing, but having, maybe different locations of the host, but the host is same, right?
The host is playing a significant role, I would say. The host is playing a significant role in terms of inducing inflammation. So the lower amount of pneumolysin or no pneumolysin or non-pore forming pneumolysin would actually induce extremely low amount of inflammation. And that would actually result in a lot less influx of neutrophils and macrophages and all that. Essentially, that would lead to lower amount of the bacterial killing. Okay. So inflammation of the host side is playing a huge role on the bacterial existence, where it will exist and how long it will exist.
Because when you are generalizing it, say for example, or comparing it with mycobacteria, so maybe there will be a situation where will be revertants, say for example, which is not producing, then it will start again producing and it will attack or it will affect the host.
So absolutely correct. So we do not know whichever bacteria is transmitting from one individual to another individual of this particular strain, whether there is a revertant mutations on that or not. We do not know about that, but that is quite a possibility.
So any other question from any of
yeah, there are a few questions from the participants, so I'll just ask. Okay, so there is Ashwin from IIT Delhi. So his question is, does this pneumolysin only affect lung parenchymal cell or also the lung macrophage cells?
So it affects any cell types present in the lung, both alveolar macrophages, type 1 epithelial cells present in the lung, as well as type 2 epithelial cells. Now, surprisingly, we see the bacteria is preferentially present in the type 2 epithelial cells in the lungs compared to the type 1 epithelial cells, which are primarily responsible for the gas exchange. But we do see the bacteria being present in the lung alveolar macrophages also. Yes. But pneumolysin, or that matter Streptococcus pneumonia, which is known to also cause septicemia, and is shown very recently by a group in University of Alabama that pneumolysin causes cardiomyocyte damage. So bacteria, while circulating in the blood, and that is typically the signature of sepsis, whenever it's circulating in the blood, it actually goes into the heart and causes heart damage. This particular bacteria under septicemic condition.
Okay. Another question from one more participant, Selena Patel from NIT Rourkela itself. So her question is the role of pneumolysin in biofilm formation is separate from the hemolytic activity responsible for tissue damage during pneumococcal disease?
It's an amazing question. I wish I could know the answer of it. So biofilm formation is a very complex process, and we are working on it. Nobody knows the role of pneumolysin in this. What we presume, and we do not know yet, that there is this high amount of pneumolysin producers actually is responsible for inducing excessive inflammation at the nasopharyngeal region during the initial phase of biofilm formation. And that excessive inflammation is required to kill off the competition from the already existing microbiota. And if that happens, then only pneumococcus can get a playing field and multiply and form a biofilm. But this is an assumption. We are trying to prove it. Hopefully we will be successful. And that is the reason possibly, on the trail of that question, that is the reason possibly why pneumococcus has this heterogeneity in pneumolysin expression while we are even growing it in vitro in a test tube.
Okay. Some two more questions we have. Okay, this is one Sevati Selvaraj from Periyar University. She's asking, is pneumolysin the only virulence factor? I think that you answered in between. Anyway, go ahead.
So that is not, any bacteria will have multiple virulence factors, not only one, because, just like we have multitasking ability, they also have multitasking abilities and all that. So for Strep pneumoniae, as per se, they produce millimolar amount of hydrogen peroxide, which is very unusual. But this hydrogen peroxide that they produce is required to kill off, again, competition from the existing microbiota. And that hydrogen peroxide, whatever it secretes outside, that kills the competing microbiota. And whatever DNA or the other thing that comes out because of killing of this competing microbiota, pneumonia is naturally competent. That's what Griffith has done, the experiment. That's why he could prove that DNA is a genetic material. Pneumonia takes up those DNA, uses up those DNA as a nutrient, or sometimes takes up that DNA and actually does some homologous recombination in its genome. And that's why you could see so many variants of pneumococcus, approximately more than 100 serotypes in the nature. So yes, it produces lots of virulence factors apart from pneumolysin. Among one of them, there are two primary virulence factors. One is this pneumolysin and one is this hydrogen peroxide.
Okay, there is one more question. Jasmine Pradhan from NIT Rourkela. So her question is, bacteria like Salmonella, after evolving, they got more virulent and evolved new mechanism to avoid host immune system. But why Streptococcus pneumoniae not following this way and try to be commensal?
Great question. I wish I could know the answer again. I love these questions. So different evolution will happen, I guess, based on the fact that what is the route of transmission. For Salmonella or Vibrio cholerae, these kind of bacteria, I think they need to cause disease in order to transmit, because they need to cause dysentery, diarrhea kind of thing in order to cause disease, right? And these are not lethal infections. Understandably, again, any infection goes beyond a certain level where you cannot control it, then it leads to fatality and all that. But normally they would cause disease because you need to go and suffer that diarrhea kind of thing. And the route of transmission is fecal oral. So from the diarrhea or dysentery, it goes from your feces, it goes to the water. And when you take this infected water, again, it can colonize your gut and all that. On the contrary, over here, pneumococcus, if at all, lungs is a very, very critical organ. And if at all it kills the human being, then it loses its home. So any bacteria, or for that matter, any living form, I would say, the primary aim is to increase as many number of homes as possible where I can live. And if at all it destroys one of the host, that means kills the host, it loses one of its home. So why it would willing to kill it? That is the question. I guess it does not want to kill it, but the excessive inflammation that the host induces in order to control the bacterial infection, that damages the host even more. And that's why the host actually suffers mortality. So it depends on what is the route of transmission, and that would actually determine the evolutionary trajectory for any bacteria in the years to come.
Okay. Thank you, sir. I think there is one more question is coming. Okay, once. Yeah, this is Avtar Mishra from again NIT Rourkela. Can pneumolysin producing bacteria be used as a vaccine, as they are easily cleared by the host, hence can develop a strong immune response?
It does. In fact, it does. It has been projected as a major vaccine candidate, particularly the non-hemolytic form. People have done some mutations in that particular toxin gene, in that particular protein, and the toxoid form of that particular toxin is actually projected as a very good vaccine candidate. It is not in the market. Till now, whatever vaccines are available in the market against Streptococcus pneumoniae is basically the polysaccharide conjugate vaccines. But that's why it's difficult, because every serotype has a different capsular polysaccharide. The composition of the capsular polysaccharide is very different. So that's why every year there is a competition between GlaxoSmithKline and Pfizer, and they keep on adding 1 or 2 capsular polysaccharides into the existing vaccinar formulation. And that's why PCV13, right now PCV15 in the market, the PCV full form is pneumococcal conjugate vaccine, and 15 stands for, that means polysaccharides from 15 different serotypes are added in that formulation. Next year, maybe GlaxoSmithKline will come with PCV17, where there will be 16 and the 17 variations of this polysaccharide will be added. So there is a competition that goes on over there. But conjugate vaccine preparations are extremely difficult, and that's why people are trying to get a protein-based vaccine which will be independent of this capsular polysaccharide. So that means serotype independent vaccine preparations, and that's why pneumolysin is a very good candidate. But no, not yet it in the market. Still far away, I suppose.
Okay. Thank you, Dr. Banerjee. I think from participant side, no more questions. Anybody from the panel in the Zoom, if have questions, can ask.
Any Professor Bhutia, you want to ask?
No, sir. Thank you.
Okay. Okay. If there are no more questions, let us congratulate again Professor Banerjee for an enlightening talk, and hopefully when we'll meet, then maybe physical reciprocal thing for the wishes, it will take place. And thank you, Professor Banerjee, again joining us from IT Bombay, and looking forward meeting you soon. Yeah.
Thank you so much. I would love to visit NIT Rourkela. In fact, one of my first PhD student was from NIT Rourkela. Your MTech batch. She has done her MTech probably from there. I could remember, but that was quite a few years back. And but she has performed phenomenally well over here. And yes, thank you so much. I would definitely love to visit, even if you do not provide me a formal invitation and all that. Definitely sometimes, yes, it will be nice.
Yeah, sure.
To all of you, and would like to meet you all of that.
Oh,
So
thank you.
Yeah, thanks. We would love to have here. Thank you, Dr. Banerjee.
Thank you.
Thank you.
So I think so. Now we'll move ahead with our second talk of session 2, and I think so. Professor Dipankar Nandi has already joined, and he's with us right now. And I think so. I will not waste much of time in going through his CV, but for the benefit of all the different participants. So I should say that Professor Dipankar Nandi was born in Kolkata, and he did his initial schooling in Mumbai. He was awarded the bachelor's degree in science from University of Bombay in 1983 and master's degree in microbiology from MSU Baroda in 1985. He performed his doctoral studies at University of California, Berkeley on characterization of epithelial tissue associated T cells, and was awarded PhD degree in 1991. He was later introduced to the world of protein degradation and proteosomes during his postdoctoral research at University of Cincinnati, Ohio. His diverse training led him to investigate 2 broad areas as an independent faculty member in the Department of Biochemistry, IISc Bangalore from 1997, that is microbial responses and host responses. He will speak today on infection-induced thymic atrophy, and we are really lucky to have him here. And we'll see what Professor Nandi will speak today. Okay, so Professor Nandi, please.
All right. A very good afternoon to everyone. Let me begin by thanking Professor Dhiman and the other organizers for giving me this opportunity to share our research interests with a wider community. So let me start off by with this slide. It shows you, this is our lab collage, and it essentially shows the different areas of interest in our lab, which can be divided up into 3 main parts. One part looks with microbial stress responses, antibiotic resistance, infection. The other 2 belong to the host response, which can be further divided up into two parts. One is role of inflammation, especially with respect to interferon gamma and nitric oxide. And the other is on the T cell part, where we have studies with T cell activation, thymic atrophy. So what I will do today is discuss thymic atrophy, which is the subject of today's talk. I hope everyone can hear me and the slides are being shared and there's no problem, right?
Yes, sir. Sir, your video is off. If you could turn on the video.
Okay, let me see. Right now, I can't see where to. Okay, start video. Okay, fine, got it.
Yeah.
All right, fine.
Visible,
okay.
sir. Yeah, thank you.
Yeah, okay, fine. All right, thank you, Vidya. Okay. So so this brings me to the subject of today's talk, which is thymic atrophy. Before that, what I will do is I will introduce some aspects of the immune system, and then I will go on to this part. Suffice to remind the broad audience that there are two main arms of the immune response. One is the innate immune part, and the other cells involved in adaptive immunity. So the innate immune cells are primarily they are the first responders. Once there is an infection, the host senses it and it responds accordingly. And you have several cells that are involved in this. Neutrophils, for example, are one of the first cells that get activated and so on, macrophages and so on. The other two are cells that are important in the adaptive immune response, and that's humoral immunity and cell-mediated. Humoral is mainly mediated by B cells, where antibodies are secreted. So the antibodies are responsible for it. On the other hand, T cells are important for cell-mediated, and that's important because what you need is cell-cell interaction. And why do you need this is something that we will see. Let me stick to a small revision of this part. So this is to remind us that you have B cells, and these, upon binding antigen, they differentiate into plasma cells and memory B cells. So these plasma cells get further differentiated into cells that generate different isotypes of antibodies of different isotypes. And you have a small population there of memory B cells, so that next time the antigen comes in, you'll be able to generate a faster response. In fact, the whole idea about vaccination is to generate the B cells, because once the antigen comes in, you are able to respond much better. Okay, T cells, on the other hand, as I said, they need cell-cell interaction. Why do you need cell-cell interaction? Is because you have what's shown over here, the T cell receptor binding to what's known as MHC molecules. So over here, the T cell receptor binding to MHC, and these MHC molecules, all of us have different self-MHC molecules, and these T cell receptors recognize our MHC molecules. And it's the MHC molecules in context with the antigens are presented, especially these are peptide antigens that are thrown up or shown on the cell surface. Once you have a cognate interaction, you have T cell activation and further events follow. And that is shown in the next slide. So primarily, there are two main types of cells. You have the CD4 positive helper cells, and you have the CD8 positive killer cells. And what is shown again is this antigen presenting cell presenting the MHC class 2 molecule over here. And this is being recognized by the T cell receptor by the CD4 positive helper cells. Once you have this cognate interaction, these CD4 positive cells, they differentiate into effector helper cells, and they are helper because they help other cells. They help macrophages, they help B cells, and in other words, they generate a much better response. So in the absence of helper cells, what would happen? So what usually happens, and this is evident in the case of AIDS patients, you can see that they are highly immunocompromised, okay? And that's the helper aspect of it, especially through cytokines, plays a very important role. The other types of cells are the killer cells, which are the CD8 positive cells. Now these, upon the cognate interaction, they differentiate into what's known as killer cells or cytotoxic T lymphocytes, which are able to kill target cells. Okay. Now, where are these cells generated? Now, this action of T cells binding to antigen presenting cells and so on occurs in the secondary lymphoid organs. So secondary lymphoid organs like our spleen, lymph nodes, the mucosal system, and so on. However, the generation of these cells occur in the primary lymphoid organs. So B cells, for example, are primarily generated in the bone marrow. T cells, on the other hand, are generated in the thymus, right? So let us discuss a little bit about the thymus. Okay. So the thymus it's located above the heart. It's a bilobed organ located above the heart, and it was named by Galen a long time back because of its structural resemblance to the leaf of the thyme. And you can see the two lobes over here, and that is why Galen named it the thymus. Okay, now let us talk. Now, this function about thymus is very interesting. Okay, and let us spend a few minutes on the person who discovered the actual function of the thymus, that is Jacques Miller. So for a long time, the thymus was thought to be a vestigial organ and which had no role. So in 1963, in fact, Sir Peter Medawer, who's a Nobel laureate and very well known for enunciating the laws of transplantation, and certainly a very big person in science, he considered the thymus as an evolutionary accident with no great significance because there was no function known. However, it is to the credit of Jacques Miller who found out the role of the thymus. And what he was doing was actually trying to look at immune responses by a virus. And this particular virus needed the young thymus or the neonatal thymus for entry and for proliferation. So the experiment that Jacques Miller set up was he removed the thymus. He neonatally removed the thymus in a group and then had some animals that were either infected, others as controls that were uninfected. Much to his surprise, he found that once you remove the thymus at a very young age, then these mice became sick. In fact, they lacked a proper, immune response. And that was surprising. And then, and this was especially surprising because if you did the same experiment with adult mice, you did not see this phenotype. Okay, so serendipitously, he had stumbled across the role of the thymus. That is, when it is young, if you remove the thymus, then you see a particular phenotype. However, he had to face a lot of criticism for these, and that's primarily because the experiments were done in what's known as dirty mice conditions. In fact, he was working in what was a renovated horse stable, so there were concerns that there were some contaminants and all that was giving him this artifact. So he had to go to NIH, do these experiments with germ-free mice, and then then people started actually believing him. So the first paper, his first paper came out in 1961. There were other researchers also who did this, who found this, but it came a little bit later. So Jacques Miller is credited as the first person to show the role of the thymus. Now, there is another interesting aspect to it. Despite the thymus playing such a critical role in generation of cellular immune cells, and T cells, as we know, are really important, this discovery was not very well recognized. And it is only last year that Jacques Miller, along with Max Cooper, was awarded the 2019 Lasker Award. And this coming 58 years after the original discovery. So you can understand. And let me also mention Max Cooper was the person who discovered B cells. So both the discovery of B cells, the discovery of the role of the thymus in T cells, these are very basic studies. And it is somewhat surprising that recognition for this has come at such a late stage. However, better late than never. So I just wanted to bring this forth, especially for students, to appreciate the role of T cells and the thymus, which is not very well studied. Okay, so the thymus is the primary site for differentiation of T cells. Now, let me also tell you that, especially with lymphocytes, the cell surface plays a very important role. And it's on the cell surface that you have different markers. And you can follow these different kinds of cells using different markers. And that is precisely what is shown over here. So the cells start off as double negative, which means they lack CD4 or CD8. Then they gain CD4 or CD8. And that's when they are called double positive. It's at this double positive stage where there is a lot of selection going on, and ultimately a small population either becomes CD4 positive or CD8 negative. Let me also show you a cross section of the thymus. What is shown is, you will see that majority, this is the H&E stain, majority of the cells are in the cortex, and these are the double positives. There are very few cells in the medulla because most of the cells die off at this stage because they are unable to be selected. And it's from the medulla that the few cells that come in after further selection, selective agents, they enter the periphery. Okay, now what would happen if you did not have a thymus? Okay, so I always use the slide to wake up my students, and I'm sure you must have had a good lunch and all, so you might be feeling sleepy. So this is a good slide to wake up, because what you see on one hand are these mice that are your normal bulb C mice. On the other hand, you have these mice that lack fur, and these are known as nude mice. They are known as nude because they lack fur, but more importantly, they also lack a thymus. Because they lack a thymus, they are immunocompromised and they accept grafts. So this becomes very important, especially in tumor immunology. You can take cells from humans, put it on nude mice, and you can grow small little cancers. And you want to see the effects of different drugs. The nude mice are especially very useful. If you did the same experiment with normal mice, the tumors wouldn't grow because they would be recognized as foreign and would be killed. So very important aspect comes out in this slide. Okay, the primary cells that we study are thymocytes, but thymocytes are not the only cells. You have other types of cells over there. You have the thymic epithelial cells, you have the dendritic cells, the macrophages, and so on. Right, but what happens is when we squeeze the thymus, primarily the cells that come out are the thymocytes, and those are the cells that we studied. But you know, there are other types of cells that are also there in the matrix, and those are somewhat difficult to study, but they're still there. Okay. Let me come to the point of education, right? I think it's very important for all of us, since we are all in education, you know, in institutes. There is stringent selection of thymocytes over here. So what is the selection for? T cells recognize self-MHC. I mentioned this early on. So the T cell receptor recognizes the MHC. And the MHC, these are R-MHC. So there has to be some selection for T cell receptors that recognize self-MHC, right? So what happens is, what the thymocytes come in, they rearrange their T cell receptors, and then you have two types of selective agents that are operative. One is positive selection, where you're positively selecting for those T cell receptors that can recognize self-MHC. That becomes very important. Now, post this, you have what's known as negative selection. However, because if these T cell receptors recognize the MHC with very great affinity, they might be autoimmune T cells. So you don't want that. So you are first selecting positively for T cell receptors that recognize self-MHC. Then you're negatively selecting for those T cell receptors that don't bind MHC very strongly. So this is, you know, a very stringent criteria, and very few, you know, less than, you know, about 2 to 3% of total thymocytes are able to satisfy this criteria and egress out into the periphery. Okay, let me also mention that this aspect about selection is not unique to the thymus. It exists everywhere, especially in educational institutions. So what are we as faculty looking in terms of students? We are positively trying to select for students. And what are the traits that we are looking for? We are looking for students who are intelligent, hardworking, you know, go-getters, will finish their studies, you know, quickly, move on in life. What are we negatively selecting for? We don't want students, you know, who are lazy, who will stay on forever, you need to move on. You don't have to be, an unwanted guest. So, so both traits are being selected for, both positive and negative. It's something very important that students need to keep in mind. Okay. So this one, in essence, shows the life history of a cell. So you have progenitor cells from the bone marrow. They enter the thymus, and it is in the thymus where I mentioned all these education activities go on. They are, and once these are selected, very few of them, they seed into the periphery. And it's in the secondary lymphoid organs where some T cell receptors may bind to the cognate MHC antigen complexes and get activated. Then they'll proliferate. And then what happens is majority of them, after the antigen is resolved, they will die, but a few small amounts will live on as a memory T cell pool. Okay. Now, the type of immune system that we know and appreciate, which is with lymph nodes, spleen, thymus, and so on, this occurred in what is known as the jawed vertebrates during evolution. Okay, so this is the type of immune system that we know of. However, there is also a smaller group, which are the jawless vertebrates, where you don't have this particular type. Okay, where you don't have, they don't have a spleen, they don't have a thymus, and so on, but they have some other equivalents. And the type of receptors that they have are very different from what we know as the B cell receptors, the T cell receptors, and so on. But even over there in the jawless vertebrates, you have what's known as structures like thymus, which are known as thymoids, okay? And they express markers that sort of resemble that of the thymus. So it's a very important aspect. And again, it's something that we need to keep in mind. All right. So there are several articles that students might want to refer to. Quite a lot of them have been written by some of my students in Resonance. And there is also a series of lectures in NPTEL, which were given by Professor Manjunath, Anjali, and myself. And students should look these up in case they are particularly interested. So with this, I will stop with my teaching aspect of the lecture, and then get on to the subject of today's talk, which is thymic atrophy. What happens is the thymus regresses or atrophies with age. And this is known as thymic atrophy, very well studied. Okay, so what happens as people age is the number of T cells remain the same. But unfortunately, what happens is that diversity is reduced. And you might have seen in your own families that people who are a little bit older often tend to fall sick and so on, because their immune system is somewhat more compromised. So thymic atrophy is known for quite some time. Okay. And this is a wonderful slide which shows exactly how that happens. Okay. So when one is born, a newborn, for example, you have a nice bilobe structure, and you can see, most of the cells generating large amounts of thymocytes. And again, even when you're 10 years old, you're able to generate. As one ages, okay, what happens is you have a lot of fat generation. And you can see the fat taking over more and very less of what's known as thymic tissue, which would actually generate thymocytes. Okay. So this is very well known. Okay. But in addition to age-related thymic atrophy, you have other agents that also induce thymic atrophy. So for example, malnutrition, stress, cortisol levels are high, so on, right? These cancer chemotherapy, infection, you have different stimuli which will also lead to thymic atrophy. In fact, these are some examples that give you this. Okay, you have obesity, for example, you have calorie restriction over here, you have the inflammasome over here, and so on. But the one that caught my attention was this. What people found out is astronauts, for example, once they go to space and they come back, they find that their thymus has atrophied more than the control age group, the ones who are on Earth. So why is that? People found out that actually, once you're an astronaut, you're up in space, you're all by yourself and so on. It increases stress levels, and perhaps higher amounts of cortisol are leading to this. I thought this was somewhat of an interesting observation, and I thought I would share it with you all. Okay, so this is what I wanted to mainly discuss. Once in kids, for example, they have nice big thymus, okay, and you have good thymic output. In fact, it's 30 to 40 grams at puberty in humans, and this is required because you need to populate your T cell pool. As you age, you have a good surviving pool, and there is low thymic output, but it doesn't affect you greatly. However, there are two conditions when this gets affected. One is during chemotherapy. And you remember, most of the drugs, kill proliferating cells. In the thymus, you have a lot of proliferating cells. And these drugs are unable to distinguish between the normal thymic proliferating cells and the cancerous proliferating cells. And what happens is as a result of which, you have a lot of immune cells that are dying. And you need to repopulate this. So, so this is why patients who are undergoing, cancer chemotherapy and all are more immunocompromised. The other, of course, are infections. And a good example, I had alluded to this, was HIV, which affects the CD4 positive cells. So your CD4 positive cells go down, and your thymus is not optimal enough, so it's not able to repopulate. So you need help. Okay. So as a consequence of this, there have been studies, people are studying on how to boost thymic function. Okay, what is shown is as you move from a healthy to an atrophied thymus, you need help, and you have different triggers that are responsible for this. And so consequently, people are looking at different drugs and different agents that will boost thymic functions. So, quite a lot of them is there. Let me mention zinc, for example, is an important one. Zinc is important because it's an important cofactor for a thymic hormone known as thymulin. So exogenous amount of zinc helps in that. You have growth hormone also, which helps, and so on, right? So infection-induced thymic atrophy has been studied for quite some time. You have different viruses, different bacteria, different parasites that cause what's known as infection-induced thymic atrophy. And it's also a dynamic process. As long as the infection is there, you have a thymic atrophy. The moment it goes back, the thymus is able to regroup and able to come back to its original size. Okay. So several years back, we decided to set up an infection-induced thymic model, and what we decided to do was to choose the Salmonella typhimurium model. So for people who are not so familiar, Salmonella is a gram-negative flagellated bacterium, and the oral of transmission is orofecal, which is you ingest it. There are two main types. One is typhi, Salmonella typhi, which causes enteric fever, and that typhoid is something that we are all familiar with. You also have a group known as the non-typhoidal Salmonella, where typhimurium, for example, belongs to that, where if you ingest it, it will cause irritation, but it will not cause typhoid. Why will it not cause typhoid? That is because it cannot breach the intestinal barrier and move on to the other parts. Okay. However, typhimurium cannot do it in humans, but it is able to do it in mice. And so that is what we have used. We have used a mouse model over here. But before going to our model, let me discuss some important aspects with respect to typhoid. Okay. One is the rise of antibiotic resistant strains. You can see over here, H58 is taking over. It's a very important one, and it's antibiotic resistance, and so it's a major problem now. The second one is what I had discussed before, which is the importance of non-typhoidal strains. And if you see in the next in this figure, it becomes more apparent. Typhoid is mainly a southeastern disease, Asian disease over here. You can see India and the Southeast Asian countries primarily being affected by typhoid. Mortality is low because these are very sensitive to antibiotics and so on. Now with typhimurium, the situation is somewhat different. Over here, you can see primarily the continent that is affected is Africa. And in Africa, what you have is a double whammy, where you have typhimurium being present and HIV. And because HIV causes a depletion of CD4 positive cells, and there are some important CD4s that are important in resistance to typhimurium. And if they are not there, you have a double whammy of both HIV and non-typhoidal typhoid, okay, which results in very high mortality and which is a major problem now. Finally, these days, we are very familiar with the term asymptomatic, okay? And we asymptomatic, and I always worry, every day I'm coming into IISc, some of my students who are staying here, they are always worried whether I'm an asymptomatic person bringing in COVID-19 over here. Students here, as Vidya would know, are very sharp and very sensitive to all these aspects, okay? So, but the original asymptomatic carrier was this person known as Mary Malone, and she was known as Typhoid Mary. And it is because she was a cook, and she would, wherever she would go, it would seem that typhoid would follow her. But she herself did not suffer from typhoid. Okay, so then that was a big surprise. But what it did was she spread typhoid, and ultimately, a lot of people got infected, and people ultimately found and isolated her. But this of asymptomatic. Okay, and so this is again something important for students, and especially with the current atmosphere that we are living in, the asymptomatic carrier needs to be highlighted over here. All right. Okay, so as I mentioned, the model that we use is a mouse model of typhoid, where we feed in quite a high dose actually of Salmonella typhimurium into mice through the oral route. And then we can do, and then we can study different aspects. So this is what is shown over here. You have a valve C mouse over here, and this is being fed through the by what's known as oral gavage over here. And then you can take a look at different time intervals. You can dissect out the mesenteric lymph nodes. You can dissect out the thymus, which is on top of the heart. You can do a variety of studies, and then it's okay. So this is one of the first experiments to be shown over here. So with time, you can see this is day 2, day 4 post-infection. The number of colony-forming units increase in the mesenteric lymph node as well as in the thymus. You will note that in the mesenteric lymph node, the colony-forming units are higher than in the thymus. However, if you look at the cell numbers actually in the mesenteric lymph nodes, they drop with infection. It's about a 2-fold drop. However, in the thymus, the drop is even greater. It's about a 10-fold drop. And this is a mesenteric lymph node. You can see with infection also they shrink a bit. But this shrinkage in terms of atrophy is a lot more pronounced in the thymus. Okay, so I'm going to try and summarize, most of whatever I'm going to present is published. So I will try and summarize some of this infection, and those of you interested should look up the papers. So what we are trying to say is that in the absence of infection, you have lots of cells. You have thymocytes, you have stromal cells, you have the other types of cells that are there in the thymus, but that we don't analyze because we take out mainly thymocytes. And it's most likely typhimurium grows in these other stromal cells, but it kills off, and this infection kills off a lot of thymocytes. Okay. And perhaps it's mainly due to the induction of host factors. And the reason we say this is because in the thymocytes, if you really plate the thymocytes and look for CFU, there is very little at all, right? So most likely it's in the stromal cells that Salmonella typhimurium is. You can do this experiment with heat-killed bacteria, but heat-killed bacteria don't show thymic atrophy because there is no real infection as such. Okay, so thymocytes are depleted. What we also found is a lot of immature cells being depleted. And so this becomes important because the mature cells seem to be more resistant as compared to the immature cells. And so this suggested a strong link between thymic differentiation and sensitivity. This was our first hint of this aspect. Okay, so what are some of the mechanisms involved? We've done these studies with mice lacking fast-Fas-cell, but there is no effect over there, which means the fast-Fas-cell pathway don't seem to play a role. What we showed is stress-induced cortisol plays an important role, and this we showed using RU486, which inhibits this particular pathway. And the other pathway which was important is inflammatory cytokines, and this again we showed using mice that lack interferon gamma. And this is again a summary of it, that there is a part role of cortisol and a part role of inflammatory cytokines, both of which they play an additive role, and they increase death of double positive thymocytes. Now, this death is not fully, doesn't account for 100% of rescue. You have other pathways also over here. But as I said, that's why there are some other pathways that we have not been able to figure out, but certainly these two play major roles over here. Okay, subsequently, we also showed the role of a stress-induced junk kinase, which plays important role over here. So if you have a junk inhibitor, you are able to get rescue. And this is all part of the idea was to identify pathways that play an important role in thymic atrophy. Okay. Now, as I mentioned, what we found out that what we observed was you always had some amount of cells that were still there. So these cells were apparently more resistant to death. So now you have a vast population of cells that are sensitive to death. You have a minor group of cells that are resistant to death. And so the idea was to find out what's the difference between these two. And so the approach again would be to use flow cytometry in conjugation with specific antibodies. And as I said, thymic differentiation is well studied, but we addressed it with respect to thymic atrophy as to what happens over here. Okay, so how do we study this? And for this, we use the fluorescence activated cell sorter. So the first cell sorter was actually built in the Los Alamos National Lab in New Mexico. Some of you might be familiar with the Los Alamos National Lab, which was important in generation of the atom bomb. And it's over here that they do a lot of other studies also. Mark Fullermeyer was the one who was responsible for this, but he used a modification of the Coulter. Coulter, the machine, is used for counting cells, okay? So he used it for sorting over here. So every time a cell goes through, there's an electrical disturbance and that gets noted, and then you are able to sort it. Okay, so this was a principle that was primarily used. This was modified by Herzenberg at Stanford University. What he did was he used a light source as made use of fluorescence or a light source for a basis of separation, right? And in fact, this and along with monoclonal antibodies, and this has greatly revolutionized this particular field. And what is shown over here is the FACS2, which was one of the first FACS that was built, and it is now in fact kept in the Smithsonian. Because it's played such a key role, and now we can't think of research without facts. Okay, so the other important thing that Herzenberg did was that he did not just publish papers and just keep it to himself. He got together with a company which is Becton Dickinson, and they wanted to commercialize this machine. And at that time, the company thought maybe we'll sell a dozen or so. But what happened is you had the advent of HIV/AIDS, and people wanted to know their CD4 counts, as a result of which the sales of FACS greatly increased. So now FACS is not only important in phenotyping our immune cells, it's also important in phenotyping cancer cells. It plays a very important role in especially finding out what type of cancer cells. And if you know what type of cancer cells, then perhaps you can modify the drug therapy specifically for this. So it's really very important. So all students must be familiar with the importance of FACS, monoclonal antibodies, and how it has revolutionized our particular field. So let me just explain this a little bit. What is shown over here is a single color, and we can do a double color, which is two different dyes, which were two different monoclonal antibodies that can be used over here. Now, what we have done is you could do a single color. This is thymocytes with CD3. You can see different populations expressing different amounts of fluorescence. You can do a two color over here. You can show CD4 and CD8. Again, vast majority of cells are the double positive expressing CD4, CD8. Now, each of these populations you can gate on, and you can look at third color. In fact, what you could do is you can do a fourth color and so on. So you could do multiple studies on this. And in fact, that's what a student of mine did. I'm going to summarize that part of the work because he went up to 6 to 7 colors of thymocytes in the absence and in the presence of infection and phenotyped it. And so let me just give you a flavor for this particular work. Again, in the interest of time, I'm going to summarize this a little bit. And so what is shown over here is looking at the double negative population, which means the cells that don't express CD4, CD8. And you can see over here, because thymocytes are very well studied, there are, based on two different markers, CD44 and CD25, you have 4 different populations over here. Now, with infection, it appears that majority of the other populations are affected, as a result of which you have an apparent increase in the DN1 population. Okay, so the cells come in, but they seem to be stuck over there because the other cells are not there, right? If you take a look at the double positives, again, you can break them down into DP1, DP2, DP3. With infection, you see primarily that the DP1 and the DP2 are affected, and the DP3s are not as much, but they are also affected, but not as much. Okay, so remember, as with thymic differentiation, the cells are becoming more resistant. The more early on, they are more sensitive, but at a later point, they are more resistant to it. This is a wonderful, this is a classic one, according to me. This is looking at single positive, and this is single positive CD8s. Now, single positive CD8s, you have 2 populations which you can separate based on CD3 and CD24. You have a population over here known as the ISPs, or the immature single positives. So they express low amounts of CD3 and reasonable amounts of CD24, which is the marker. Now, the mature population expresses higher amounts of CD3, which is T cell receptor. But now what the student was able to do was separate it based on CD24. So it's this population which is the more immature, and this population which is more mature. Now, under normal conditions, you are unable to see this population much. However, with infection, you're able to see this. You know, now there's an accumulation over here. And the ISP population is gone. So he's done kinetic studies to show that the ISP population is really the earliest population that vanishes. That is the population that is most sensitive to infection-induced thymic atrophy. So very, very interesting. And the accumulation of this population is there with infection. So people knew thymocytes. There's a reduction in thymocytes. But what we showed over here is not only is there a reduction in thymocytes, there is also a slowing down of the differentiation process. So this would be like saying, if you're looking at a highway, you see cars going at a certain speed. However, now if you put a block in them, what you've done is you've reduced it. So you will now be able to see it in a much more slower manner, and that is exactly what has been done over here, and that is why we are able to pick these up. Okay, again using high-end flow cytometry for this purpose. Okay, so again, this is a summary of it. The DN population is less sensitive, as was mentioned. The CD8 positive SPs are also less sensitive. However, the ISPs are one of the first ones to be lost during infections, and DP1, DP2, which is the double positives, are also lost over here. And this is a summary of the data that he generated. This is suffice to show that the DN1s are the less sensitive ones over here. And you have different thymic populations. And you can see he's also identified a population that is very sensitive to interferon gamma. Okay, so again, using mice that lack interferon gamma, he was able to identify this population. Okay, so again, these are published studies. So I would, those of you who are interested should take a closer look at it. Okay, so once we had this part done on infection-induced thymic atrophy, one of the questions was, what happens if you use different kinds of thymic atrophy? Okay, because infection-induced thymic atrophy is not the only type. There are other kinds, as I mentioned. So what we did was we compared infection-induced thymic atrophy over here with other types of thymic atrophy, one of which was LPS, or lipopolysaccharide or endotoxin mediated thymic atrophy. All right, and we compared this with two other kinds. One is the etoposide mediated one, because etoposide is used in the treatment of cancers. So what would happen? It again causes thymic atrophy. And then you have a dexamethasone induced thymic atrophy. Now, dexamethasone is an anti-inflammatory. In fact, it's inexpensive. It's often used for treatment of autoimmune diseases and so on. But, you know, cortisol has its own issue, or dexamethasone has its own issues, because steroids have also some other issues, but it is still used because it's inexpensive. Okay, so we standardize these different kinds of thymic atrophy, and then then try to see what happens. Okay, so this is what we found. The etoposide affects all thymic populations, as does the lipopolysaccharide. The infection-induced thymic atrophy affected primarily the DN, ISPs, and the double positives. The dexamethasone surprisingly affected primarily, on the other hand, primarily the more mature populations over here seem to be affected by this. Okay. What are some of the mechanisms? Antioxidants are thought to be important over here. There have been a lot of reports of antioxidants being important over here. So we tried to address this with N-acetylcysteine. And this again becomes important because a lot of people take antioxidants in the hope of improving immune function and so on. What we found over here is that N-acetylcysteine was effective in rescuing thymic atrophy in 2 out of the 4 modes that we had studied. One was infection-induced thymic atrophy, as well as lipopolysaccharide-induced thymic atrophy. So both the infection inflammatory were one part of it, but it had no effect on etoposide and the dexamethasone. So this becomes important because, when people study, they usually look at their favorite form of thymic atrophy, and then they say, oh, it rescues thymic atrophy. But what this study showed is that we should not generalize. Some of the drugs and all might be useful in some forms of thymic atrophy, but not others. So this becomes important over here. So these are some of the highlights. I'm sort of winding down right now. The changes occurring in thymocyte subpopulations were studied. We found roles for glucocorticoids and interferon gamma in modulating different thymic populations. Compared thymic subpopulations to other modes of thymic atrophy, and we identified N-acetylcysteine to identify thymic atrophy during both infection and inflammatory or LPS-induced thymic atrophy. So we need to see what happens with infection with an attenuated strain. Remember, the model that we have is an acute model, right? All the mice die, within a certain set of time. What would happen if some of the mice survived? Okay, at a later point in time, what would happen? Okay, especially in a chronic scenario, what would happen? What are the other mechanisms involved? Can other additional compounds be identified? And more importantly, can these be tested to improve T cell numbers in a clinical setting? I think that is really the important part over here. Okay, so let me just thank the people who have done it. So most of the data that I showed was started by a student of mine, Mukta, and it was followed up. In fact, all the differentiation part has been done by Shamik over here. And this is, I think we had gone for a lab trip to Avani Hills near Kolar, and it has very nice rock formations. And I would urge everyone over here to take a look and go for a hike once the situation becomes normal. Then this was the current lab up in January, but of course there's been a big disruption because of the pandemic. Almost everyone went home except one poor student over here who gave me company throughout the lockdown. We will have to see, because once the students come back, there'll be a lot of changes, unfortunately, because the pandemic has been disruptive. So we'll have to see how to get going post this situation. I'd like to thank again Mukta starting it off. Shamik did most of the work. Supriya has done this work with an attenuated strain, and I've not presented her data. Hopefully, we will be able to present this at a later point in time. Abhinay and Subhashish helped in the study. Vashisht Adhika played a very important role. Vashisht is very good with analysis of flow cytometry data. These are not very easy to analyze and interpret, so he was very helpful. Sanmoy and Taru have been playing a supportive role in all this. I'd like to thank the divisional facts facility as well as the animal facility, without which we wouldn't be able to do anything. I thank SERB and the DBT IISc partnership program, and this is our lab symbol. Some of you might recognize Geronimo Stilton, and we seem to have a lot of similarity with that. I'd also acknowledge the Department of Biochemistry. The department was started in 1921, and we were supposed to have our centenary celebrations, but that has also been affected because of the pandemic. And I thank you all for your attention. Thank you.
Thank you, Professor Nandi, for a great talk. And I take this opportunity to thank you for taking your precious time and address the gathering out here. And I can understand the situation right now. Pandemic, it is all over. The institute.
Yes, that's
same.
Correct. Yeah.
And being head of the safety security over here, so I think so. We were also following the SOPs, but IC was thinking of bringing back the students. So I don't know how the situation now is. Many students, whether they have come back or not.
No. Some have come back, but a lot are not, because cases in Bangalore are also increasing. So it's a little bit of a tough situation right now.
All right. For us also, actually, we are devising the SOPs, and also maybe in future, we will also be thinking in that same line, because without students, it's definitely all the labs. It's the same situation everywhere. So I can also understand. And Professor Patra is already has unmuted himself. So Professor Patra, please.
Hi, Dipankar.
Yes. Hello, sir.
How are you?
Good.
Obviously, we heard your lecture. So I have only one question. How many mouse you sacrifice per year?
Oh my God. I stopped counting. Don't even bring it up. Now nothing, zero.
Sir, if we count that, that make us to feel guilty. Oh, how many we killed. So we don't count them.
That's right.
It's okay. It's nice. Student level presentation. Very good with data and others. So I don't have any specific question. And honestly, I don't work in this area. So I don't have that much expertise also. But yet typically, the T cells, as you are talking about.
You can stop sharing your slides.
Yeah,
sir.
sure. I'll stop. Yeah.
Okay,
That's what I wanted to ask. Yeah.
Yes, sir.
T cells.
Yeah.
As you are the master of that one now, right now. So how can we use these T cells? Can be used better way for cancer treatment?
Oh, for cancer treatment, I think a Nobel Prize was given on that, especially with the checkpoint inhibitors. In fact, I urge you to, we published a review on T cell co-stimulation, checkpoint inhibitors, and anti-tumor therapy. Please take a look at it, because that gives quite a detailed role on T cells and how they affect cancer cells. So let me just explain it in a few sentences. So if you see, usually what happens is when cancer, there is a fight between the tumor cells and the T cells, and often the tumor cells win, right? And because the T cells, the tumor cells are smart, they are producing different factors which sort of suppress T cell responses. Okay, so now what has been done is, if you can some way boost T cells, and that is what was done using the checkpoint inhibitors, and if you're able to do that, then you skew it in such a way that that the T cells now start winning and that the tumor cells lose. But despite all this, the efficacy, if you see among patients, is about 30% or so. And so therefore, there have to be other strategies that you need to couple with to try and boost up this response again. right? And in fact, so we have, that review it would be a good one, in fact, because I think, and that would really help. So I urge you to take a look at it. But in general, people are looking at different ways by which you could boost T cell responses against tumors, because there's a big fight usually going on. And as I said, in most cases, previously also the tumor cells have been winning, but now we are thinking of ways or by which we can boost T cell responses.
Okay, thank you.
Sir, may I ask a question?
Sure.
Yeah. Sir, this is a very naive question, but have you checked for the balance of Th1 versus Th2 cells with respect to typhoid? How?
No, so we have not. We have not. You see, Sandhya, because our experiments are, they usually, because it's an acute model, and so by day 4, day 5, the experiments are over. So it's very early on to have a T cell response as such, the T cell response that we know of, right? So when people look at T cell responses, what they use are mainly attenuated strains and all. So it'll go on for a longer time because it takes some time to generate the T cell response, right? I think there are lots of people who have worked on T cell responses on it, and that's what they use. In our case, we have not done that. Plus, our focus was the thymus over here, right? So that's why we have not looked into that aspect of it.
Okay.
Anybody from the panel have question? Okay, so thank you, sir. Thank you very much for the nice talk. I have one question, and there are some questions from the participants also.
Sure.
The question mine is like this atrophy. Would you expect similar kind of outcome when we compare with the different kind of pathogen rather than just only the salmonella or any other pathogen?
Yeah, that's a good question. So our reason for using salmonella was, by the time we started, that would be an easier model to use to ask this question. Okay, and but certainly, in fact, the NAC part is actually what we are trying to get at. Is that this is where, in fact, we try to broaden it to other forms of atrophy to ask whether they work or not, right? And that's why we found that, some of these work only on some, but not some of the others. Right? And so it's a useful model, but one shouldn't stop over there. One needs to, which is why I tried to say in the future, would try and find, other parts, and ultimately it needs to get into a more clinical sort of a setting, because everybody is interested in trying to boost up thymic function.
Right, sir. Okay, so we'll go, sir. Thank you, sir. So we'll go some of the question from the student side. So there is one question from Satyanarayan. So he's asking, any viral infection, okay, very simple question, sir. Any viral infection, interferon is formed. Which cell of our body or cell organelles responsible for the secretion of this? So very naive question is there.
Right. So interferons are produced by a wide variety of cells. You have different types of interferons, and they are produced by a wide variety of it. So interferon gamma, the one that we are talking about, they are made primarily by T cells and by NK cells. Small amount is also made by macrophages, right? And it's usually produced upon activation. So T cells have to be activated in order to produce. Yeah. So those are all important parts.
Okay, so another question from Ashwani from IIT Delhi, sir. His question is, do we have any change in type of T cell, like T helper or T cytotoxic cell development as the thymic atrophy advances?
Okay, so what happens with thymic atrophy? The cells that are lost are primarily the immature cells, right? You can see the mature cell loss is very little. In fact, that is best reflected when you take a look at the lymph node data, where there's a small drop. And the reason why we haven't looked at Th1 versus Th2 because it's a short model, and the mice die by 8 to 10 days of infection. So it's very early to look at those type of responses. We've sort of focused mainly on the thymus, and over there the Th1 Th2 doesn't come up as much. But now if you have a chronic model and you will have some cells that survive on at much later, then maybe one could consider taking a look at that. And so that's a good point.
Okay, so another question from Selina Patel from NIT Rourkela itself. With age, due to atrophy, there have been report of T cell maturation in the liver and intestine. So is it true? If yes, how the positive and negative selection of T cell occurs there?
Okay, so the primarily way by which it occurs is the thymic one. Okay, so there might be small parts, that may be able to contribute, but those contributions are very little. So the primarily responsible ones are over here, and there is a lot of genetic data. There's a lot of model data to tell us that in fact the thymus is responsible. So if you just take a look at the nude mice, for example, if you had other sources, for example, they should have been more immunosufficient, but they are not. They are primarily, so the thymus is primarily responsible for it. So if that is not there, then it becomes a problem. And you also have other models in humans. For humans, for example, who have problems with thymic function and all, they are primarily immunocompromised.
Okay, so another question, sir. This is from Mosami Das, again NIT Rourkela. So is the mechanism of depletion in ISP is same in all mode of thymic atrophy?
Okay, mechanism of ISP depletion in all modes. Good point. We haven't seen that. We haven't addressed that part of it. What we have found that they're the most sensitive cells. So we use that as actually a marker to try and say, okay, it's the initiation of thymic atrophy. But with both RU486 and with this thing and with the gamma, you see rescue. But whether detailed look into whether about particular ISPs is something we have not studied, but maybe we should take a look into it. It's a good point.
Okay, so there are a couple of more questions, sir. So we'll be taking. So very similar question, Jagannath Pradhan from NIT Rourkela. So is there any relationship between thymic atrophy and the type of infection? I think that you just answered also.
Yeah, so you have it is not restricted. There are other modes also, and we have looked at some modes, and so it becomes important.
Okay, I think one more last question we'll take, sir. This is Ashirwad Behra again from NIT Rourkela. So IL-6 impart protective immunity during the salmonella infection and is also responsible for thymic atrophy. So does salmonella infection mediate thymic atrophy by host factor like cytokines?
Yes. In fact, that's what we showed with the gamma knockout mice. In fact, we showed the role of host factors. So it's primarily host factor driven. Even the cortisol is produced as by ourselves, right? So it's host factors that are responsible primarily for thymic atrophy.
Yeah, I think no more questions, sir. So thank you very much for a nice talk,
sure,
sir.
my pleasure. Thank you so much for the invitation. It's a pleasure.
So we would love to have you here again sometime, sir.
Yeah, sure.
So
once the situation
stabilizes, last time,
right, sir.
Yes. I enjoyed very much. I remember that.
Yeah, unfortunately, I was not there, sir, so I didn't have chance to host you that time.
Sure. I'll be more than happy. Okay. Anytime.
Sure.
I think so. From on behalf of our department and institute, we thank Professor Nandi in coming on board as far as this lecture is concerned. And
thank you so much. Thank you so much. My pleasure.
Thank you.
Okay, thank you, sir.
Thank you very much.
Memento is due.
So now I think so we'll move ahead with our third talk of session 2, and this talk will be delivered by Dr. This talk will be delivered by Dr. Sandhya Marathe, and she is from Pilani. So if you go back to her education, so I think so most of the work she has done in IISc. So she did a bachelor's from University of Pune before going to MS and completing her PhD from IISc Bangalore in Department of Microbiology and Cell Biology. And then she stayed there for a couple of more years as a research associate, and after that, she made a move to Pilani, where she was a visiting assistant professor for almost like 3 to 4 years, and joined as assistant professor starting from April 2017. And we are really fortunate that on a short notice, Dr. Marathe has agreed to give talk in this platform. And I think so overall our department as well as all the presenters, they will be really eagerly waiting to listen about, her talk, which is on characterizing the role of CRISPR-Cas system in Salmonella. So Dr. Marathe, please.
Are you able to see my screen, sir?
Yeah, it's fine.
Thank you, sir, for your kind introduction. So, first, I would like to thank Dr. Rohan and Dr. Vidya for inviting me for this conference and giving me an opportunity to present my work in this platform. Okay, my major focus is on Salmonella pathogenesis, and in today's talk, I would be discussing about characterizing the role of CRISPR-Cas system in Salmonella. So this work is done in past 3 years by two of my research scholars, Nandita and Simran, with the help of two ME students, Ankita and Krishnaveni. So without them, this work wouldn't have been possible. That's why I start my talk by acknowledging these 4 students. So before we start discussing about the role of CRISPR-Cas system in Salmonella, let me first give you a brief introduction about CRISPR-Cas system. And I guess most of you would be aware of it by now, that what is CRISPR-Cas system and its role. So CRISPR-Cas system, it stands for clustered, regularly interspaced, short palindromic repeats is for CRISPR, and Cas is for CRISPR associated genes or proteins. Okay. So this system it has two components, an RNA component and a protein component, which works in coherence with each other to bring about their function. So when this system was discovered, it was discovered as an adaptive immune system in prokaryotes. So just before this talk, we heard to Professor Nandi that what is adaptive immune system and it has a role in the mammalian defense. So when this system was discovered in prokaryotes, it was a big discovery, because apart from restriction modification system, no one knew about adaptive immune system in prokarya. So how this system works as an adaptive immune system in prokaryote. So when you hear about adaptive immune system, immediately one thing occurs to your mind is memory, right? So how is a bacteria recalling the memory of previous infection and then counteracting the subsequent infection by the same pathogenic element? And here the pathogenic elements are mobile genetic elements, which comprise of plasmids and phages. So now in this diagram, you see that this phage is infecting, and as it is infecting the bacteria, it is ingesting a DNA, its DNA, or else it is ingesting a genomic content, which can be DNA or RNA in the bacteria. Now this genomic fragment is processed by the bacteria with the help of the proteins encoded by Cas genes, and it is incorporated in a loci called as CRISPR loci, which is made up of a leader sequence highlighted by blue, repeats, as the name suggests, short palindromic repeats, and these repeats are interspaced by a fragment which is named as spacer, but this fragment is essentially from the mobile genetic element. So this is where the memory comes into picture. The memory of the previous infection is stored in the form of a DNA fragment, which is acquired from the genome of the phage or from the plasmid, right? So now what happens when the bacteria is growing normally? This CRISPR loci is transcribed into a CRISPR RNA, or it is called as pre-CRISPR RNA, which is then further processed with the help of Cas proteins to produce small CRISPR RNA, and this small CRISPR RNA represent one spacer and two repeat sequences. So this repeat sequence form a hairpin loop. Now the Cas proteins recognize this particular structure and bind to it and use this spacer as a guide to go and bind to the invading mobile genetic element on subsequent infection, right? So now there are different CRISPR-Cas systems present in bacteria. The one which is widely studied is type 2 system. So in this system, there is one more element which is important for silencing the mobile genetic element or for destroying the mobile genetic element. What the element is, it is called as tracer RNA. So this tracer RNA, why it is called as tracer? Because it traces the CRISPR RNA. So this tracer RNA binds to the direct repeat, and now this pre-CRISPR RNA is processed into CRISPR RNA, and now the other CRISPR proteins are brought to this assembly, and these CRISPR proteins have multiple properties. Some of them have an ability to bind to RNA, some of them have an ability to bind to DNA, and others have a nuclease and helicase activity. So in the type 2 CRISPR-Cas system, Cas9 is the nuclease, and that is it is a huge protein which takes care of all the activity, that is binding to the nucleic acid, performing the helicase function, that is unwinding the double-stranded DNA if it is present, and then allowing the base pairing of the spacer with the target as it is shown over here, right? Whereas for another type of CRISPR-Cas system which has recently been studied in past few years, which is type 1 system, instead of one single huge Cas protein, it has multiple Cas proteins which play a role in this activity, where different Cas proteins form a cascade complex along the target DNA, and the CRISPR RNA helps, okay, and the CRISPR RNA forms the complementary base pair with the target DNA sequence. And once this is formed, the Cas is recruited to the site, and its nucleic acid can be DNA or RNA. Have I lost the connectivity? Hello?
No, it's fine. It's fine now, Sandhya.
Yeah. Okay. Right. So now the target nucleic acid can be DNA or RNA. For type 1, it is DNA, and for type 2, it can be DNA as well as RNA. So once this is done, once the target is been chopped off by nucleic acid, nucleases, the mobile genetic element is rendered inactive and it won't cause infection in the prokaryote. This is how the system works. So one thing which I would like to highlight here, and which I would like you to remember, is that the binding of the CRISPR RNA, which is represented as guide RNA over here, to the target RNA has to be 100% complementary, so as to bring about the degradation of the target nucleic acid. If it is not 100% complementary, the degradation won't happen. Okay. But now afterwards, people have shown that even if there is no complementarity, it has a role. What is the role? We'll discuss later. So, as I told, type 2 CRISPR-Cas system is widely studied, and it was initially studied as an adaptive immune system of prokaryote. But as people started studying it in detail, they also found that this system regulates the endogenous genes of the bacteria, thereby regulating the bacterial physiology. And what do you mean by physiology? Different aspects of bacteria like biofilm or bacterial virulence, right? Now, as scientists understood the mechanism of this CRISPR-Cas system, they tweaked the system so as to edit the genome or to gene edit for gene editing. So if you just go in PubMed and give the keywords Cas9 and gene editing, you will find 6,000 plus 6,000 plus articles which are citing the use of this CRISPR-Cas system for genome editing. So this CRISPR-Cas system is the highlight of today's science or today's research. It is widely used across all the kingdoms, right? But the type 1 system is less studied, and recently people have started elucidating the mechanism of type 1 system. So people have also use this type 1 system for gene editing. So now if you give keyword Cas3 and gene editing, Cas3 is a signature protein of type 1 CRISPR-Cas system. You see only 9 articles which cite the use of Cas3 for gene editing, right? So you know now this is very clear that the type 1 system is very less studied. So what is known about type 1 system? It is known how it degrades the incoming genomic DNA or RNA of the phage. The biochemistry of the nuclease, Cas3 nuclease is known. People have also studied the regulatory mechanism of this CRISPR-Cas system, and there are hints of this regulatory RNA which is produced by this system in regulating different endogenous genes which may have role, for example, in biofilm formation and RNA processing, thereby impacting the physiology of the prokaryote. So now this particular system is present in our well-known or loved bacteria by all of us, especially from molecular biology point of view, E. coli. E. coli, this system is being studied, and they have also a potential regulatory gene bacteria, which is Pseudomonas. So the second article talks about the regulation of virulence in Pseudomonas by the CRISPR-Cas system, and this is also a type 1 CRISPR-Cas system. Right? Now, Salmonella is a close relative of E. coli, and today you have got a good dose of Salmonella infection in the morning from Professor Deepshikha Chakravarti and in the evening by Professor Dipankar Nandi. So they have made my job easy. So now the question is, you would be thinking why she is studying CRISPR-Cas system only in Salmonella, not anywhere else, right? Vidya also asked Dipankar sir that why he chose Salmonella to study thymus, role of thymus or thymectomy and all those things. So why Salmonella? Again, Dipankar sir has explained much about it, but to give few more aspects, throughout the world, 14.3 million cases of Salmonella are reported, of which 1.5 lakhs die annually, right? And as highlighted by Professor Nandi, that Salmonella infections are prevalent in Africa, Southeast Asia, and also in Central Asia. And the complications are majorly due to the drug-resistant strains, which are in Southeast Asian countries as well as in Indian subcontinent, and this is a major challenge. So this poses a challenge, it is a challenge for researchers like us to look for novel drug targets so that this particular pathogen can be eradicated from India, or at least we can control it. So now, as discussed in last few minutes, CRISPR-Cas system regulates the endogenous genes and bacterial physiology is known, but it is known in type 1, type 2 system, and it has just begun to be elucidated in for type 1 system. Right? Salmonella has type 1 system, so we are targeting it. We want to study the CRISPR-Cas system in order to see if it could be used as a novel drug target, or we can design therapeutic strategies based on it. Okay, so this is why Salmonella and why CRISPR-Cas system of Salmonella. Okay, so now you also heard about typhoid Mary, right? She was an asymptomatic carrier. Why was she was an asymptomatic carrier? She was an asymptomatic carrier because, okay, sorry, I think I lost that slide. Because there was a biofilm formation in her body by Salmonella, and that was causing recurrent infection. Okay, so biofilm formation is important for Salmonella pathogenesis. Right. Now we study, or we discuss that the CRISPR-Cas system acts via its spacers and Cas, right? And the spacers have to have complementarity with its target so as to perform its function, right? So now with this concept in mind, we looked for whether the CRISPR array or the spacers show any complementary binding with the endogenous genes, that is, whether it is having any self-targeting spacers, right? Now, Salmonella has two CRISPR arrays, CRISPR1 and CRISPR2, and one Cas operon to carry out its function, right? So what we did is I took the spacer sequence out from this CRISPR arrays and tried some in silico analysis to find the match of the spacer with the endogenous genes, and we found significant spacers being targeting the endogenous genes. So we started shortlisting it and categorizing it. How did we categorize it? So we know DNA is made up of a template strand and a non-template strand for transcription, right? So now, whatever targets we found, so based on the binding of the spacer to the template targets as transcription or translation, right? And if it is not binding to any gene but to the intergenic region,
Okay.
it
I
will
think
be
Sandhya, we are
classified
getting a
as
connection. I think there is some
intergenic.
problem from
Here
your
is the stats for
network issue is there.
I think so. There is some network problem with the Marathe.
targets for the strain.
Dr. Marathe, there is some problem with the internet.
I think we lost the connection. Sandhya, hello.
Can you change the connection to other system or something like that?
No, not yet, Sandhya. It's not
Sandhya, it's
not audible properly.
Yeah, it's not audible. It's not working. It's not working. I think so. She's not there right now. Vidya, can you contact her?
I'm talking to her. Ron.
Otherwise, actually, Dr. Shabir Ahmed is also there. So who is next in line? So just wait for another couple of minutes. So we will just make it clear. So Dr. Marathe is again joining back. So let us see.
She's joining again, Ron. So we'll see.
Yeah. So there is some problem, I think so, at her side with respect to
from her side, the network issue is happening.
Yeah. So it is not working.
Sandhya, can you hear us? No. I think we will stop this for now, Ron. So we can see later on. We'll go for next talk, no?
Okay, so you please talk to her, and
I'll
then
just
we can
talk to
think
her.
of. Yeah. Okay. So Dr. Ahmed is, I think so. We'll just ask Shabir then. Is that fine, Vidya?
Yes.
So you please talk to her, Vidya, because I think so. There's some severe internet connection issue over there. Is there any separate internet port or some other provider? Vidya?
Rohan, are we going for next talk or will we wait? We're going for
No, she's already joining back again.
one more time. She's trying. So if it works,
So
fine.
this
Then
is the same. This is the same port, or she's using
same only. They have been using it regularly, but that problem has never happened. So this is today. I don't know what had happened. So
Okay.
let's try once. So we'll see if it works. Fine. If not, then we go for the next talk.
Okay.
She's back.
Are you able to hear me?
Yeah.
Yeah.
Now
Now it's fine. Hopefully now it doesn't get disconnected.
I hope so. Actually, we use the same connection for maybe. Yeah.
Yeah. Okay. Let's continue. So we'll see. Hopefully.
I was saying that we found the endogenous gene targets of Salmonella and classified them into three categories: translational, transcriptional, and intergenic, based on the binding of the spacer to a template strand or a non-template strand. Right? And now when we analyzed this target, we found that some of these targets were involved in two of the important mechanisms of Salmonella, which is biofilm formation and pathogenicity. So now Salmonella alternates majorly between two lifestyles: biofilm when it is in the environment and the pathogenic when it is inside the host. Right? Inside the host also it forms biofilm on the gallstone. As I told you that typhoid Mary, who was an asymptomatic carrier, she had biofilm on her gallstone formed by Salmonella, right? And this was the reason for recurrent infections in her. Right? And now, when the bacteria is in the biofilm, it is recalcitrant to the antibiotics or any antimicrobial agent in that matter. Right? So, we focused on the bacterial biofilm formation and role of CRISPR-Cas system on the bacterial biofilm formation by Salmonella. Right? So now, in order to study the regulation of Salmonella biofilm by CRISPR-Cas system, what we did is we generated the knockout strains of CRISPR-1, CRISPR-2 array, and the Cas operon. And we tested the ability of these knockout strains to form biofilm at different time points. Why different time points? Because when the biofilm formation starts, it initiates slowly and it matures progressively, right? So in order to mimic this maturation of biofilm, we studied biofilm formation by Salmonella at different time points. That is after 24 hours, after 48 hours, and after 96 hours. Surprisingly, what we found was that the knockout strains of CRISPR-Cas system, they were defective in the biofilm formation in the initial phase, which is the attachment phase of the biofilm. And as the time progresses or as the biofilm matures, the biofilm forming ability of the knockout strain was higher than that of the wild type strain. Right? So we concluded that the CRISPR and the Cas knockout strains show biphasic biofilm formation when we compare to wild type. That is, in the initial period, it is less than that of the wild type, and in the later phase, it is greater than that of the wild type. Okay. Now, we also analyzed the biofilm architecture. So when the biofilm is formed, it is like a society. It is a three-dimensional structure which the bacteria form, and there are water channels and channels for nutrients to be supplied to different parts. So this architecture was studied with the help of scanning electron microscopy, and we found that the knockout strains show distinct biofilm architecture than that of the wild type, and also they have more of a coating around the bacteria, and generally this coating is of polysaccharide layers, right? And if you closely observe this biofilms, you would see that the CRISPR2 knockout strains and the CRISPR1 and 2 double knockout strain have similar architecture. What does this signify? This signifies that CRISPR2 acts upstream of CRISPR1. Okay, so this was a very interesting result. But what was worrying us is what could be the reason for the biphasic or biofilm formation by the CRISPR and Cas knockout strains. So we dwelled deeper into the mechanism of biofilm formation, and we found that the different biofilm components are important at different phases of biofilm formation. For example, appendages like flagella, curly, and pili are important in the initial phase, whereas the exopolysaccharides like cellulose, eDNA, or LPS are required in the later phase of biofilm formation. So we started looking for this components, and what we found that, okay, and we started looking at this component and got answers to our questions. So in the initial phase, we found that flagella is important. Now flagella is important for the motility of the bacteria. So we tested whether the knockout strains show compromised motility or altered motility. What we found is the strains indeed showed compromised motility compared to the wild type, and this compromised motility was reverted back when we supplied the CRISPR1 and CRISPR2 genes in complement, suggesting that whatever effects we were seeing were not the side effects of the mutation process. Right? So now why the defect in motility? Very simple answer, there could be due to defect in the flagella. So when we looked for the flagella around the bacteria, we found that the knockout strains had lesser flagella compared to the wild type strain, and this was due to reduction of expression of flagellar genes like the FlyC as shown by the Western blot analysis here, and expression of other flagellar genes shown by real-time PCR. Okay? So now flagella, I told it is important for the motility. So how this is related to reduced biofilm formation by the knockout strains in the initial phase? See, when we inoculate, biofilm formation is done under static condition. So we inoculate and keep this tube stationary and observe for the biofilm formation. So due to gravity, generally the bacteria would settle down, and if they have motility, they would move to the periphery at the air-liquid interface, at this interface, and then attach themselves, forming a thin biofilm ring, which is a initiation of biofilm formation, right? Now, as the flagella is less, the knockouts migrates slowly to the air-liquid interface, and flagella is also required for attachment. As less flagella, less attachment. Hence, less biofilm formation in the knockout strains. Then the next question was for 48 hours. How at the late after 48 hours, the biofilm formation is increasing for the knockout strains. So what is important? I said LPS and cellulose. So we started studying the LPS profile of the wild type and the knockout strains. So we isolated LPS from these strains and ran a phage gel and looked for the LPS profile by performing staining. So now LPS is made up of different components like lipid A, core oligosaccharide, and O antigen. So we say that there is difference in the O antigen and the oligosaccharides with the knockout strains when we are comparing to wild type. So now this could impact the cell-cell interaction, cell surface interaction, and development of external appendages by the bacteria, and also the surface tension. And why this? Because LPS have role in all these functions, right? So with this, we could explain the altered behavior of the knockout at the later time point. Now we tested for cellulose. When we tested for cellulose, we found that yes, knockout indeed forms more cellulose, and this was also evident from the crinkling pattern of the biofilm in the knockout strains when compared to wild type. Okay. So for the later phase, the CRISPR and Cas knockout strains have altered LPS and showed increased cellulose production, which correlates well with the increased biofilm formation in the later phase. So to summarize, in the initial phase, less of flagella, less of movement of bacteria from the bottom to the air-liquid interface, and less attachment, hence less biofilm formation. Later on, more of cellulose, which increases the thickness of the pellicle and also retains the moisture. Retaining of moisture helps in easy supply of nutrients to different parts of the biofilm, keeping the bacterial cells viable and hence inducing more of biofilm production. Right? This is how we studied how CRISPR-Cas system is impacting the biofilm formation by Salmonella. So now the next question is whether this regulation of biofilm formation by CRISPR-Cas system is true for all other servers of Salmonella. If you remember, the speakers before me, this told that there are more than 2,500 servers of Salmonella. So will this results apply to those servers as well? Luckily, we had a paper which was released in January 2020, where they found that the CRISPR-Cas system is in fact enhancing the bacterial biofilm function. That means the knockout show reduced biofilm formation, and what we observed was knockout is showing increased biofilm formation, which was contradictory to what we observed. Right? So the fact that CRISPR-Cas regulates biofilm formation is true, but what kind of regulation is distinct for different servers. So this was done for Serover entritidis, and we are working on Serover typhimurium. Right? So now we also looked at whether these CRISPR spacers are conserved across the species and servers, and if there is a conservation, then they would have similar regulation of the genes. When we looked at Salmonella enterica and Salmonella Bombay, we showed that there was no sharing of spaces, whereas different servers of enterica, some of them have sharing of spaces as it is highlighted over here. So darker color means they have sharing, but not all servers share the spaces, but only few servers. So if we try to characterize these servers deep, we see that, okay, only few servers, like for example, here Serover typhimurium and Heidelberg, they share significant amount of spaces with each other, but not with other servers, right? So the CRISPR spacer is not shared across species, but across some servers of Salmonella enterica. Okay. So now what is the scene for strains within these servers? So the scene is, yes, there is conservation of spaces for strains within the server. So here the dark color indicates 100% conservation of all space, 100% conservation, and lighter color or whiter color represents less conservation. So if you look, some servers like Heidelberg, Agona, they have significant spaces conserved among the strains of themselves, whereas some servers like typhimurium, the strains show variation in the conservation of spaces. So this means that different strains would behave differently in terms of their physiology with respect to CRISPR-Cas system regulation. Right? So now can we correlate this to the host range of Salmonella? What I mean by that is there a correlation between the CRISPR-Cas system and host range, the characteristics of CRISPR-Cas system and host range? So if you see in this map, the spacer map, that some servers like Gallinarium, Dublin, Pularum, Typhi, and Sendai, they have very few spacers. So now, can we correlate number of spacers with the host range of the server? To some extent, yes. So like servers Gallinarium, Paratyphi, and Typhi, they have fewer spacers, whereas servers like Typhimurium entritidis, over here you see Typhimurium, it has good number of spacers. Similarly, entritidis have good number of spaces, and they are host generalists. That means they can infect multiple species. Yes, there seems to be a correlation between the CRISPR-Cas system and the host range. To analyze deeper, we studied in the phylogenomics of CRISPR-Cas system in Salmonella. So now, as told in previous slides, that the spacers are not that conserved. So in order to study the phylogenomics, we focused on the leader sequence of the CRISPR array and the Cas operon. So when we analyzed the CRISPR1 leader sequence and Cas operon and studied the phylogenetics, what we found that the serovers got segregated into two distinct clades. Right? This is one clade and this is another clade. So and in this clade we had signature serovers typhimurium, Salmonella serover typhimurium, which is widely studied, and serover typhi getting segregated into two distinct clades. Thus, we label this clade as CRISPR1 STM and CRISPR1 STY. STM stands for typhimurium, STY stands for typhi. So in both the CRISPR1 leader and Cas operon phylogeny, we find two distinct clades, and these clades are consistent for both the trees, right? Now, again, when we looked deeper into it, what we found is that the typhoidal serivers, they grouped into the CRISPR1 STY clade. They also had some broad host range serivers, but these serivers are frequently associated with human infections. Right? And then we also had non-typhoid and other serivers which are non-typhoidal, as explained in the previous talk, that non-typhoidal ones, they do not cause typhoid-like symptoms, they only cause gastrointestinal disease. Okay? So they clubbed together, and the broad host range had multiple spacers, whereas the host adapted had fewer spacers, and host restricted, that means they infect only primates, they had fewer spacers. Right? So there was a correlation between the spacer content and the host range. So now but we were surprised when we looked at the phylogeny of CRISPR2 leader. We did not find two distinct clades. In fact, the CRISPR2 leaders tree was to some extent similar to the MLST tree, which signifies the genome evolution. So we have Bongori, which is a different species from Salmonella enterica, and all serovers of enterica, they club in one clade, and Bongori comes as a second clade. In MLST tree also, we have Bongori as a separate clade and other serovers in one clade. But the branching of these, the Salmonella enterica clade, do not match. So is this a result to worry? So we took it as a challenge. No, we should not worry. We should think broad. So when we looked at the CRISPR-Cas evolution, which was reported by other strains, it is known that the CRISPR-Cas system could be possibly acquired through horizontal gene transfer event. So now we wanted to look for the signature of this horizontal gene transfer in Salmonella. And when we looked for the signature genes like the transposase, nuclease, helicase, what we found was the CRISPR-Cas system is flanked by the transposase gene. So this transposase gene could have been responsible for the lateral transfer or horizontal transfer of this CRISPR-Cas system into Salmonella. And to support this, we also check for the GC content. So we know that difference in GC content also signifies horizontal gene transfer event or lateral gene transfer. So the whole genome is 52. That for CRISPR1 and CRISPR2, it was high, which again supports that the CRISPR-Cas system is acquired through horizontal gene transfer. So we next thought whether this system, the CRISPR-Cas system, is shared with other species of Enterobacteriaceae family. And yes, we found that yes, it is shared. And when we generated a phylogenetic tree of the CRISPR1 leader, what we found that the typhi strain, the typhi serover clubbed with E. coli, whereas the typhimurium serover, it clubbed with Klebsiella pneumoniae and Shigella boydii, right? That means there is horizontal gene transfer happening among these species. But here the interesting point to note is, here also the typhi serovers and the typhimurium serovers segregated into two different clades, but even though they belong to one single subspecies. So it is Salmonella enterica subspecies enterica serover typhi and serover typhimurium. Even though they belong to same subspecies, they are clubbing with different genus altogether, right? So this again signifies that this is a horizontal gene transfer event. So this is about CRISPR1. What about CRISPR2? So when we looked for the CRISPR2 leader in other Enterobacteriaceae family, we did not find any match, right? So now when we analyzed this entire result from an evolutionary angle, or we looked at the evolutionary pattern of the CRISPR-Cas system, that is a CRISPR1 and Cas together and CRISPR2 as a separate unit, we came up with a Salmonella, before diverging from the other Enterobacteriaceae species, it acquired CRISPR1 STM type Enterica and S. bongori. They segregated into two different groups, or they segregated from each other. Am I audible?
Yes, Sandhya, go ahead.
Yes.
Continue.
No, I had an error, that's why, yeah. So they acquired CRISPR2 and then there was a divergence, and hence Bongori had a different CRISPR2, that it was falling apart in the CRISPR2 tree. And after the divergence of S. enterica and S. bongori into two different species, some serovers of Salmonella enterica acquired this CRISPR1 STY type CRISPR-Cas system, right? So after that, it got segregated into two clades. The STY clade, which clubbed with Citrobacter and E. coli. Sorry, E. coli I have by mistakenly put here. It should be here, right? And this is how we talk about the evolutionary pattern of the CRISPR-Cas system in Salmonella. So we began studying whether it is having a role in endogenous gene regulation and physiology. And when we started extrapolating it to other serovers, we landed up in defining the evolutionary pattern of the CRISPR-Cas system in Salmonella. So with this, I end my talk. And before I stop my presentation, I would like to acknowledge Pilani for providing me a platform to work here and initially providing me grant to initiate my lab. I would like to thank SERB DST for funding me to carry out this project, which I discussed with you. I would also thank NIT Rourkela for giving me an opportunity and platform to discuss my work. I would like to thank my collaborators, Dr. Vidya Negi from NIT Rourkela, Dr. Chandrajit Lahiri from Sunaway University, Malaysia, and Dr. Franklin Nobrega from University of Southampton, UK, for their valuable inputs during the work and brainstorming discussions when we discuss our collaborative research work. Thank you.
Thank you, Dr. Sandhya Marathe, for giving us opportunity to listen to your enlightening talk. And once again, thank you very much to agree on a short notice, because we really planned it on a very short period of time. So thank you very much again on board. And let me again ask all the speakers or our faculty colleagues who have joined through the Zoom platform that if you have any questions, I guess there will be plenty from our HODs, Sir Professor Subhajit Das, because he also works on biofilm and all. So any questions from anyone who is joining us from the Zoom platform, and then we will ask the questions from the students or the participants who are joining us from the YouTube. And I think so Ashwini sir has also joined. Ashwini sir, do you have any question?
No.
Although you joined late. Yeah. So Vidya,
nobody understood the talk, or they understood it very well.
No. Actually, the thing is this CRISPR-Cas, like when you were saying like these are the contradicting reports. So means it will be like a serovar or a species specific, like what you want to say.
To some extent, yes. With Salmonella, at least we are seeing it is serover specific description of this CRISPR arrays.
Okay.
We are yet to look into different species. For example, in E. coli, different strains of E. coli, how it behaves. But
Okay.
for Salmonella, definitely we find that there is the spacer content varies from serover to serover.
So what is the relation with the pathogenicity of these particular civil wars?
Okay.
With respect to
Relation of
CRISPR-Cas?
CRISPR-Cas system?
Yeah, right.
So, see now, as I told that the spaces were matching with the endogenous gene. That means they were forming a complementary base pairing. And now it is forming a complementary base pairing, even though it is not 100% base complementary. But the cascade system goes and latches on it. So if the cascade latches on it, it could interfere with the expression of the gene. Right? If it is latching on the genome, it could interfere with transcription. If it is latching on mRNA, it would interfere with translation. And now, if these endogenous genes are virulent genes, then it would impact. And there is a paper in bioRxiv, which is not yet published in a journal, but they have found that yes, there are this cascade complex going and binding to the genome loci. And when we analyze this, there were virulent genes picked up and also some biofilm forming genes picked up through this interaction. And hence it is regulating. Now, if the spacer content varies, it might regulate the same gene because the spacer is just 20 plus nucleotides long, and whatever base pairing we see, it is up to 11 base pairs or at times up to 10, 15 base pairs. So, and the gene being a longer size, so it could regulate similar genes. But as I discussed that Serover entritidis and typhimurium, they behave very differently when the CRISPR-Cas system is knocked out. And they do not share a single spacer with each other.
Yeah. Hi, Sandhya. So as you're saying with the species specific also, no, as the host specificity they have, they are showing the differences.
Yes.
So in typhimurium case, what you have done so far, when you're correlating with the biofilm formation, the next step was with respect to infectivity in the in vivo system or in host system or in cell line system. So any comment on that part, like how
Yeah,
will be?
when we are knocking out this CRISPR-Cas system, we see attenuated virulence of the bacteria. The bacteria do not perform well inside the host. Or we have tested it only in C. elegans and cell culture system. There we see an attenuation in the virulence of the knockout strains.
Okay, so from the panel, if there is no question, there is a question from the participant side. There is a question from Mausumi Das from NIT Rourkela. The question is that apart from the impact of biphasic biofilm formation, what are other aspects of the knockout strain of CRISPR and Cas in comparison to the wild type?
One is reduced pathogenicity compared to wild type. We have also looked at from the enterobacterium, which is the siderophore aspect. We see that the strains show variation in the acquisition of iron from the environment because there is difference in the production of siderophores by the knockout strains, so which would impact the survival of Salmonella in the environment as well as in the host. Inside the host also, Salmonella needs siderophores.
Okay. And another question we have from Jagannath from NIT Rourkela only. So his question is, what is the function of tracer RNA and pathogen associated motif, PAM?
Tracer RNA is not found in Salmonella in type 1 system, but in type 2 system. But there is no direct association between tracer RNA and PAM because PAM is recognized by Cas proteins, not by the tracer RNA. But tracer RNA helps in pairing of CRISPR RNA to the target nucleic acid. So that is important.
Okay. Another question we have, this is from, Avtar Mishra again from NIT Rourkela. The question is, can CRISPR-Cas be used as a host-directed therapy to interfere with the virulence gene expression of pathogen?
Yes, actually, this is the second aspect which I am looking into, and for this I have collaborated with Dr. Franklin Nobrega, where we are trying to use endogenous system of endogenous system of Salmonella to target itself. So how can we tweak Salmonella to target its own genome and degrade its own genome such that it does not survive? So instead of using antibiotics, we can use its own system to degrade itself. yes, it is possible. And people have done it with type 2 system, and they have tested it with, I think, Staphylococcus. I'm not that 100% sure, but they have tested this. It works.
Okay, so we have another question. This is from Lincoln Naik from NIT Rourkela again. So the question is there any change in the size of genome of organism when a protospacer is added? Up to how many number protospacer can be added? Does it change the characteristic of the organism?
Okay, yes, it changes. For example, in one of the serover, which is serover 10SA in our study, so it has 63 spacers. It has 63 spacers which have been acquired. And so we do not, as of now, know what could be the limit, but up to 63 it can acquire. But there is another study with serover Montevideo where they have looked at different strains of this serover, and there they found an interesting observation is that if the size of the CRISPR array is short, then the genome size of that those Montevideo strains is larger. And if the array size increase, the genome size is less. And this they correlate with the mobile genetic elements. They say that if you have more of spacers, these spacers would target the incoming mobile genetic elements, degrade it, and not allow the integration of incoming mobile genetic elements into the genome. So yes, there is a correlation.
Okay. I think from participant side, there are no more questions. So thank you, Sandhya. I thank you for accepting this very short notice invitation.
Thank you for inviting me, and I'm sorry there was problem in my internet and we had to stop in between. And later on, I got panicked and I hurried up. I hope you understood the.
And that's okay. At least we could join back, so it was fine.
So I think so on behalf of NIT Rourkela and the Department of Life Science, I must congratulate Dr. Marathe for giving the enlightening talk, and hope so that we'll meet you soon, because what I heard and during our lab meetings also, I think so your name has been discussed a lot many times, because generally we conduct our regular meetings with Vidya. So hope so that you will come down and to Rourkela, and then we can have some good interactive sessions. But whatever is the demand of the time right now, so we are using the virtual mode, and hopefully we'll meet soon. Thank you, ma'am, again on board, and thank you for coming. Yeah, thank you very much.
Yeah, thanks, Sandhya. Thanks.
Now we will move with our last speaker of the day, and it is my again great privilege to introduce the last speaker, who is also good friend of mine. We were together for our PhD in Institute of Microbial Technology, although his supervisor is right now in IISc Mohali. So actually Shabir completed his PhD in biophysics and biochemistry from INTEC, and he joined later Beckman Research Institute. And before I think so his research profile was also good, and that's why it took him to City of Hope in California as a postdoctoral fellow, and where he continued his protein structure function work to understand the cause and mechanism of neurodegenerative diseases caused by mutations in dynein and dynectin mutaprotein. And I think so later in the same organization, he joined as a staff scientist also in drug discovery and structural biology core physics, where he worked mainly on structure-guided design of small molecules and anti-cancer drug screening. So since after coming from our spillover was almost like at the same time when we were in US, and I came to join in THSTI. He was in US, and after 2 years of THSTI stay, then I moved back to NIT Rourkela. And at the same point of time, he joined THSTI. And over there, he joined as, I think so, senior scientist in HIV vaccine translational research laboratory. And I think so since last year, he's senior research scientist in infection and immunology in THSTI Faridabad, and he's working on structure-guided protein engineering work to design, characterize, and validate the immunogens as potential vaccine candidate and in the area of antibody as a diagnostic and therapeutic for infectious diseases. And I think so it is really a great privilege for every one of us to go through what right now, Shabir is doing over there. And I'm really happy for him and thankful for him for accepting our invitations. And everyone is looking forward to a great talk from Shabir. Yeah, sir, please.
Thank you, Rohan. Can you hear me?
Yeah, sure.
So thank you, Rohan, for the nice for the invitation and for the nice introduction. So I am going to share the screen now. Okay.
Yeah, you can share.
Okay, so you can see it now.
Yeah,
that's fine. So the title of my talk is the protein subunit based immunization design in HIV, and so basically the thing is that HIV is a retrovirus that causes AIDS, and it attacks the CD plus T cells and depletes immunity. And there are approximately 36.7 million people, this is a little old data, are worldwide are living with HIV and AIDS, and there is a lot of casualty every year. And with lot of effort and lot of interventions, still the HIV prevalence is quite high, and particularly in the sub-Saharan Africa. So there is a need to tackle this disease, and probably a vaccine is the best approach, because vaccine can eradicate the disease from the completely eradicate the many diseases. But designing an HIV vaccine is really challenging. So this is a structure of the HIV HIV virus, HIV-1 virus. So the only the exposed protein part on the HIV virus is the surface envelope glycoproteins, and so these are only exposed antigen. And this is also the binds with the receptor on the cell surface, that is CXCR4, and enters the cells and infects the cells. The problem with the HIV is that once a person is infected, he is infected forever, because the genome gets integrated into the DNA of that person. So there is actually no functional cure. The only way is that how to keep the viral load low, so that no more and more T cells get infected, and there is low level of T cells that other opportunistic pathogens take this opportunity and causes a major disease. So in HIV life cycles, the first it binds to the CXCR4 with the GP120 envelope glycoprotein. Fusion happens, and the uncoating of the viral genome takes place. They get integrated in the genome after converting into the cDNA, because these are retroviruses, and then they take over the cellular mechanism to produce its own viral particles, and then gets assembled, and then they keep on producing more and more viruses. If HIV is a quite old disease for now, almost 30 years, and for every step there is some kind of interventions are available now. So like for anti-inhibitor that block the interaction between the CXCR4 and the GP envelope protein, the reverse transcription inhibitor that inhibits the reverse transcription of the RNA to DNA, and there are integrase inhibitors, and then there is a protease inhibitor. The reverse transcriptase inhibitors are the most successful for now, and most of the people who are taking reverse transcriptase for the daily basis are keeping the viral load is very low. But then why the need of the HIV vaccine? Because even with the current trend and with the current problem, if we continue...
1 minute. Can you increase the microphone volume, or somehow can you speak a little bit loud, because sound is...
Okay. Can you hear me better now?
Yeah, it's fine.
Okay. So the problem is with the HIV is that even if with the current intervention we continue, this is a green line. The disease will not go down. But if we increase the interventions, there is a high chance if there is a rule that is called 90-90-90. So 90% attachment to prevention, 90% attachment to taking retroviral drugs and all these things. Even with this purple line, the cases will go down up to about a 0.6 million by 2070. But if a vaccine is introduced in the meantime, assuming 70% efficacy, this will drastically, even with the current trend, drastically reduce the disease burden. So there is a need for HIV vaccine, but really this is a challenging problem for designing a vaccine. But and the only option in HIV is to design a protein subunit based vaccine. Other forms of vaccine designing will not probably work well, and there is a chances of there's chances that it may instead of protection it may give the disease, because this is a retrovirus and its genome gets integrated, its DNA gets integrated into the genome. So if we try to use inactivated HIV-1 virus, and somehow if that gets inactivated in the body, that will itself create the infection. Killed viruses are also not good because it alters the surface antigens and does not give right kind of antibodies to give protection. So the only thing what we can do is taking the GP120 envelope protein and design it in such a way, in a soluble form, that when it is introduced as a vaccine and immunization, it will give rise to right kind of an antibody that will give protection. But this is very challenging. The challenge is that HIV has very limited surface antigens. Compared to chikungunya virus or dengue virus, they are very well decorated with the surface antigens. But in case of HIV, very limited. And the other thing is that it is a transmembrane glycoprotein. So it is in two parts, the GP120 and GP41. They are actually translated as a single chain called GP160 that gets cleaved between the GP41 and GP120, and they remain associated as a non-covalently. And that is important because if this cleavage does not happen, the functional trimer, that is a functional envelope glycoprotein, does not form. So cleavage is necessary. The other problem is the huge sequence diversity. So HIV is a reverse transcript, has a reverse transcriptase, and the estimated error rate is almost 3 × 10⁵ per bases. Based on that, there is a huge sequence diversity, and HIV is divided into many subtypes, that is A, B, and C, and D, and there is a different prevalence of that also in many areas. And so here is you can see the diversity of this done by genome analysis, and it can be compared with the diversity of HIV compared to the influenza. So this is the HIV diversity only in Republic of Congo in 1997. See how huge the diversity compared to influenza. And this is the HIV diversity in a single individual. So what happens because of the reverse transcripted, mutations are kept on introduced in the genome, and this gives a high sequence diversity. And this becomes a problem because any antibody that is neutralizing a clade A HIV may not neutralize the clade B or clade C. So a vaccine designed for clade A and is introduced, and person gets infected with clade B will not get protection. So the challenge is to have an antibody or a vaccine that will neutralize a broad antibody response that at least can take care of the different subtypes in terms of giving protection or neutralization. The only surface exposed antigen is the envelope protein. Now, this is envelope protein is again first they are very few, and the very few are also very metastable structure. This is the closed conformation, that is the best functionally correct conformation, but they may also appear as an open conformation where the 3 trimers almost falling apart, or there may be aberrant structure where there is only monomer or there's only the stump is there and only one GP120 attached to there. So many possible available conformation may be available, because and the structure is very metastable, tries to fall very fast. The other problem is that heavy surface glycosylation. So one monomer, so this is one monomer of GP120 and GP41, and they together is a trimer of heterodimer. They together form the trimer. So one of them has a almost 20 to 25 glycosylation sites. And these glycans are quite long, almost 9 manos. So taken together on one native-like trimer, almost there is 60 glycosylation sites, and they cover the whole surface. And these work as kind of immunoevasion. So they don't expose the epitopes, protein epitopes, for which the antibody may be elicited. So, but even with these challenges, there are almost 10 to 20% population, people in the population that produces neutralizing antibodies, and they are called elite neutralizer. So there are two kinds of antibodies are found. There are non-neutralizing antibodies and neutralizing antibodies. So non-neutralizing antibodies bind on those surfaces that are non-functional form, and the epitopes are not important. So even if the antibody binds with that forms, it does not neutralize because they cannot bind to the correct conformation. Neutralizing antibodies bind to the correct conformation, that is the functional conformation, and blocks its interaction with the host cell receptor and prevent infection. Now, the broadly neutralizing antibodies are the antibodies which can bind to neutralize clade A, clade B, clade C, or other clades. And there are 4 major neutralizing epitopes that has been identified so far. These are the V1, V2 apex area. There are the glycans in the V3, there are epitopes in the V3 region. So these are the V3, the whole sequence of the GP120 is divided into a constant region and variable region. Constant regions are mostly the regions which have shown less variation in terms of mutations during reverse transcriptation, and variable region shows more mutations. So this and so V1, V2 is the first variable 1 and 2 loops, and V3 is the third variable loops. Then there is a CD4 binding site. CD4 binding site is the site where the CXCR4 gets CD4 is the co-receptor that gets attached and then helps in the virus entry to the cell. And one more important region in terms of neutralization is the region where the GP120 and GP41 becomes close to the forms are non-covalently associated. This is GP120 and GP41 area. So these are membrane proximal region because this region is very close to the membrane of the viral surface. Now, first part of my talk is associated with the metastable conformation. So how to stabilize the metastable conformation so that we can take an opportunity to design a soluble form of the immunogen. So what is the soluble form of the immunogen? So this is a native-like trimeric conformation that is membrane associated with the on the virus membrane. Now, if I want it to be a soluble form, it has to be in this formation, this form where I have removed the transmembrane region. Now, removing the transmembrane region is challenging because as soon as you remove the transmembrane region, they completely fall apart. So first, so the challenge is to convert this to this conformation, and it should be soluble, it should be trimeric, and it should also be closed conformation, not a trimer of this open kind of conformation, because that will induce more non-neutralizing antibodies instead of neutralizing antibodies. Now, there are many approaches are there, and one of the best approach that work and is the SOCIP mutation. So what is SOCIP mutation? So this is GP120 and GP41. We know that it gets cleavage, but they remain associated non-covalently. So a mutation was introduced between in the C5 region of GP120 and HR1 region of GP41, that is heptarepeat 1 region, 16 mutations are introduced. So they will form disulfide bond. And one there is a mutation, that is I556 to P4, that is isoleucine to proline mutation. So SOS stands for the disulfide bond formation and isoleucine to proline mutation. Because this HR1 and HR2 are heptarepeat 1 and heptarepeat 2 region, they forms there are two conformations, that is pre-fusion and post-fusion. So when isoleucine to proline mutation is introduced, they remain in the pre-fusion conformation. Because if they go into the post-fusion conformation, they form a 6 helix bundle, only the GP41, and that helps in the entry of the virus into the cell. So to keep it to the pre-fusion conformation, a isoleucine to proline mutation is introduced to make a kink so that it remains in the pre-fusion conformation. And this mutation is introduced at the 559 at the HR1 region. So it is stabilized in the pre-fusion conformation rather than post-fusion conformation. And then it is truncated at the 664, near about 664 amino acid to remove the transmembrane region so that it remains in a soluble, to keep it in a soluble form. Now, we had a patient who was a light neutralizer, and the envelope virus is isolated from there, and the envelope were amplified from there and cloned. And there was one envelope that we identified is actually identified by Dr. Jayanta, who was at that time working at National AIDS Research Institute. And that envelope is called 42J41. And that is a subtype C envelope. And here the importance is that the subtype C, in fact, the 50% of the world population are infected with subtype C. Almost all the Indian continent and the South African continent are with the subtype C. So this is a major dominating strain. So we tried to make an immunogen from this envelope, from the envelope of this virus. And when we used the standard SOCIP mutation, it did not work. So we had to, we started thinking how then we should, what kind of stabilizing mutation we should introduce to make it work. So here we decided to make some mutations so that we took several approaches like cysteine tethering and all these things, but one of the approach was to do the domain swapping, because BG505, that is so far it is a clade A envelope, and so far most well characterized envelope because the SOCIP mutation worked really well there. So we thought that if we introduce the GP41 region from BG505 to GP120, will that work? Because then in that case, you will all have the V1, V2 epitope, V3 epitope, and the CD4 binding epitope from the GP120 of 42J41 envelope. Only the GP41 will come from the BG505. So we made three constructs. One is where we completely replace the GP41 with the BG505 envelope. We replaced only the HR2 region, and we replaced only the HR1 region. And then we screened it. So for screening, there is already very well characterized known antibodies in the field of HIV that's broadly, and these are really broadly neutralizing antibodies that binds only with the trimeric conformation. So this is broadly neutralizing, and this is trimeric native-like trimeric envelope. So when we tested the first construct, we found those antibodies, broadly neutralizing antibodies, are really binding well in the Western, but non-neutralizing antibodies are not binding. And VSCO1 is an antibody that binds with all the construct, even if it is a open conformation. So this kind of introduces a positive control. When only HR2 replace, we don't see any binding. With only HR1, we do see binding. So we came to the conclusion that if at least if the HR1 domain from the BG505 is introduced into 42J41 along with the SOCIP mutations, it will form a native-like soluble trimer to run a SDS page with or without SDS. So when SDS is introduced, because this is a cysteine link bond, you can see the GP41 is dropping here, and this is migrating little low compared to this one when there is no DDT. The other thing is that people have standardized it really well for in the native scanning electron microscopy to see the trimers. So whenever there is any envelope that is purified in a soluble form, one standard technique is to see whether they are forming a native-like trimer or not. So in the if it is forming a native-like trimer, it will appear like this. So in a trilog conformation. And these are the aberrant conformation which is not forming the native-like trimer. So since in our case, we saw that two construct with HR1 replaced, all the HR1, HR2 both replaced are binding to the neutralizing antibodies, we tested both the construct under the electron microscope, and we found that HR1 is forming trimers, but there are also aberrant trimers. But in case of when the whole GP41 is replaced with BG505, all the populations that we saw under the electron microscope are forming trilo propeller shaped native-like soluble trimer. So we decided to further characterize only this construct, and we dropped this one. Now there is one more mutation that is introduced by Peter Kong's group in the Biology Research Center in the NIH. They found that if you introduce a mutation from cysteine mutation at this isoleucine 201 and alanine 433, they form a disulfide bond here and they block the V3 loop. Now the importance of the V3 loop is here is that this region is quite flexible and they open and close and gives lots of breathing. And an antibody that is called 17B and 44752D binds this breathing structure. Now, if a structure keeps breathing, then there is a high chance that the structure is not so stable. So these mutations are introduced to make it more stable. So we also introduce the same mutation, and the breathing of this construct can be induced more in the presence of soluble CD4. The CD4 is the co-receptor. So there is an increasing titration of CD4. So when this mutation is double cysteine, this double cysteine mutation when introduced, when 20 times molar excess of soluble CD4 introduced, only then these antibodies are binding. Without that mutation, the 17B and 447 antibodies are binding. So this mutation further stabilizes the construct where the breathing of the V3 loop is also constrained. Now, the next step was to test whether this design conformation is really binding to the broadly neutralizing antibodies or not. And for that, we did biolayer interferometry. This is a kind of SPR, and we measured the kinetics. So this is a PG9. These are the panel of the broadly neutralizing antibodies, and these are the well-known broadly neutralizing antibodies. So PG9 and PGT145 antibodies binds to the trimer apex, and they are binding really well. PGT151 antibodies binds to the GP120 and GP41 region, and they also binding. We could not determine the kinetic here because KD, because here with one trimer, two antibodies bind. Here 1-to-1 binding. This is also 1-to-1 binding. But PGT151, PGT121 antibodies per trimer, two antibodies bind. So to know the affinity and KD, we actually have to use a FAB, but we do not do that. But we see a binding. So that is these broadly neutralizing antibodies. So we come to the conclusion that these broadly neutralizing antibodies are binding to this construct, but not the non-neutralizing antibodies are not binding, and even 17B is also not binding. So we further tested its thermostability, because if we are using it as an immunogen or as a vaccine candidate, it should have sufficient degree of stability so that it does not the structure falls apart. And one parameter is to see how stable it is it is sensitive to the temperature. So we did a DSC and we found that the melting temperature is almost around 73 degrees centigrade, and the same is reported for BG505. And we heat the protein kept the protein at 37 degrees centigrade for 3 days, and even after 3 days we found it is still binding to the broadly neutralizing antibodies. So basically, so here we have designed an immunogen from Indian grade C envelope that is in a trimeric native-like soluble conformation, and it's binding to the known neutralizing antibodies. So in indirect way, we are saying that it has all the epitopes that are neutralizing are in the correct conformations, but not binding to the non-neutralizing antibody. So all the epitopes that are not essential are hidden or not are not exposed. So we can probably use as a potential candidate to use in immunogenicity studies to see whether they are giving right kind of antibody response or not. Now I will come to the second part of the talk, where we try to modulate the surface glycan by creating glycan holes. Now what does that mean? I will come next to that. So glycans are integral part of this envelope protein in HIV. There are around 60 glycan moieties on the HIV envelope primer that and glycans make the half of the mass of the envelope protein. And they play a very important role in maintaining the native-like trimeric structure, because removal of glycans not from here and there cause the structure to fall apart. There is one glycan known very well, 192 or something, that has been shown that plays an important role in proper folding, but the consequences of and tolerance of glycan removal is not fully understood. And these glycans, since cover the whole of the surface, they kind of act as a immunoevasion. Now the idea here is that this is a trimeric envelope protein, and this is the neutralizing target site, and it is covered with a glycan. So when a body sees a virus, the B cells need to attach to this target site for the initial priming. But because the glycans are present, the B cells priming does not happen the way it should be. So what the idea here is that if we can remove those glycans from these sites, it will expose the target site, and this will help in better priming of the B cells. So here is the cartoon representation. So if we clean the target site by removing the glycans, initial priming will happen better, and then immunize this with the wild type envelope which have all the glycan. But since initial priming is happened, it will go through affinity maturation, and then the mature bodily antibodies may have a bodily neutralizing aspect. So here we are just trying to increase the chances or the prospects of getting neutralizing antibodies faster by giving an opportunity to the B cell for better priming. Now, here, this is a list of viruses, envelope viruses, which have been designed in a native-like trimeric form. And these are a couple of them only, given the so much huge sequence diversity and all these things. The V1, V2 loop is highly variable. So you can see the sequence differences. So either the variation is in the amino acid sequence and also in the loop length, and also presence of N glycosylation sites. So this is V1, V2 loop, and this is V3 loop. And V1, V2 apex forms an important site for an important neutralizing epitope. So we did some analysis with the same 42J41 envelope with the swap construct that I described just before. And the modeling shows that this V1, V2 apex, we are seeing the trimer from the top. So this is the top of the trimer, and these are 3 GP120. So this is the lysine-rich patch, which is completely covered with the glycans. But if we remove those glycans, these lysine green color patches opens up. So we have also shown by modeling that binding of PGT145 antibodies actually clashes with many of these glycans. Now here comes the concept of germline antibodies. Now germline antibodies have been identified in HIV patients where these are the early lineage of the mature antibodies. And it has been shown that these germline antibodies binds with a envelope with a, which have a less glycans, or the particularly known as a envelope with the glycan holes. Like in BG505, there is a patch which have a less glycan. So the idea is here to design an envelope which removing the glycan from the apex region and then see where we can improve the binding of those germline antibodies, early antibodies, which compared to the wild type. The first thing was to see whether this. So we remove the three glycans N187, N133, and N137 to open up these patches. And first we tested this construct in the screening where they are still binding to the known well-characterized broadly neutralizing antibodies or not. And we found that yes, they are binding to the well-characterized broadly neutralizing antibodies. So we purified that protein. So here the red is the glycan deleted construct and the black is the wild type construct. And this forms a trimer on the as a native page, and on the native scan electron microscopy, these are also forming a trimer. Now since we were focused only on the V1, V2 apex, so that our collaborator from Netherlands, Amsterdam, he gives us this germline PGT145 and germline PG9 antibodies and germline PGT128 antibodies. And in the BLI, we showed that there is improved binding for all these three antibodies in the glycan deleted construct compared to the wild type construct. When we tested for the neutralization, we also find a better improved neutralization potency for the glycan deleted construct compared to the wild type construct, at least for the germline PGT145, but not so well for the, not much difference we saw for the germline PG9 antibody and PGT128 antibody. Now here we have to remember one point, that germline antibodies are early precursor and so they are always weak in neutralization. So we don't know at this point whether this weak neutralization is, we are speculating this weak neutralization is because these antibodies cannot neutralize so well because they are not still mature. Next, we tested this glycan deleted construct with other antibodies which are V1, V2 apex directed, and we found that it's binding to the mature antibodies really well, as it should be, and it's as binding really well with the other. So basically, when we deleted all these glycans, it's improving the binding to the germline antibodies, but at the same time, retaining the binding to the mature antibodies, not hampering the conformation, so that it loses the binding to the other apex directed antibodies. And at the same time, we also tested whether it is binding to the antibodies for the other site, like PGT121, and that's bind to the N332 patch, another neutralizing epitope, and PGT151 that binds into the GP120 and GP41 region, PGT135 antibody that also bind to the N332 side. So not only it is retaining the glycan deleted construct, not only retaining binding to the V1, V2 directed antibodies, but also retaining the binding to the antibodies that are away from the V1, V2 sites. And we also tested its thermostability, and we found that it is quite stable with a TM of 74 degrees centigrade, almost same with as it was for the wild type that we showed earlier, 73 degrees centigrade. Oh, sorry, this is exercise. So in summary here, we stabilize an Indian peptide C envelope by swapping domain from another stable envelope. The envelope was further designed to create glycan hole to expose neutralizing epitope. The stabilized envelope binds exclusively to known broadly neutralizing antibodies and not to non-neutralizing antibodies, indicating that the design construct as maintaining native-like conformation in soluble form, and the high TM also indicating that it can be stored for a long time, or even if for immunization when we are adding the adjuvants or all these things, or even in the blood stream, it will return, it will be stable. Now I will briefly touch about some recent finding, that is isolation and characterization of SARS-CoV-2 antibody. So here we know that SARS-CoV-2 spike protein is also very much like HIV envelope spike, and fusion 1 mechanism, class 1 fusion mechanism also is the same class of fusion mechanism like HIV, and it has a 3 domain: the N-terminal domain, the RBD, and then the S2. There is the comparable to the GP41, and the NTD and the RBD region is comparable to the GP120. Now, most of the neutralizing antibodies in SARS-CoV-2 binds to the RBD only. So here what we did, we purified this RBD in the soluble form and used it as a biopanning to identify a SCFV fragment that binds to the RBD. We screened those binders, and we found one binder, that is number 62, that binds quite well in the ELISA with the RBD. We created 3 formats of that. So this is the SCFV format. We made the SCFV FC format and the full length antibody, that is with the IgG1. All the 3 forms are binding really well with the RBD. And we did also BLI to show that they are binding really well with a high KD. KD was not really with a low nanomolar, but it was around 500 nanomolar range. We did modeling, and we also showed by facts that when we are expressing the SARS-CoV-2 spike on the cell surface, they are all binding to these antibodies are binding to those cell surface expressed SARS-CoV-2 spike protein. We did modeling, and we found that this SCFV actually binds a little away so this is the RBD in the blue, and this is the antibody, and they bind little away from the S2, that is the receptor for the SARS-CoV-2 spike protein. And we also found that this is not neutralizing antibodies, but we are working on this problem to identify any SCFV that is neutralizing. Actually, we are hoping if any antibody or SCFV that binds close to this S2, that will probably block the binding to the S2, that is the receptor, cell surface receptor for the host, and will block the infection. So, and that will be a neutralizing antibody. So with this, I would like to stop here. And I would like to thank all the collaborators and the funders we have, and all our lab members and all these things. Thank you.
Thank you, Shabir, for a very nice presentation. And I hope there will be many questions from the participants. But I just want to take this opportunity to ask you, I am not very expert, or I don't know much of this field, but I think so SARS-CoV-2 and HIV, so which is I think so lentivirus, right? It's a retrovirus.
Yes, retrovirus. Both are retrovirus.
So both are retroviruses.
Yes.
So it's just a general question. People, they are saying within, say, for example, 1 year or so, that vaccine is already under a clinical trial and all those things. But in HIV, that progress is very slow. So what is the reason for that?
See, it is very difficult. So with the HIV, though both are retrovirus, the challenge with HIV is that once the genome is integrated, the person is infected for the whole life, and there is no functional cure. I mean, you cannot completely remove that fragment of the HIV from the genome. But in case of SARS-CoV-2, that is not. So for HIV, we cannot use the inactivated or that kind of virus because there is always the problem. If in any case there is a change in virulency, that person will get infected. But in SARS-CoV-2, there is a hope that inactivated virus can be used, and the Bharat Biotech vaccine candidate is the inactivated SARS-CoV-2. So that is one of the challenge. Second challenge is the native-like conformation. Trimeric native-like conformation. In SARS-CoV-2, we have seen that the RBD, even if there is single RBD, anything that can block the RBD-H2 receptor, it can block the infection. But in HIV, it has to be in a trimeric conformation in a quaternary structure. And that maintaining that quaternary structure in HIV is more challenging than in SARS-CoV-2 because also in the SARS-CoV-2, people are using only RBD domain, that is only 28 kilodalton protein, and they are getting antibodies against that, and some of those antibodies are neutralizing. Because even if you can block only the RBD, but in HIV there is no single domain that you can block and expect that it will neutralize. That is why when the HIV research started, people were saying it's not a problem. They only took the GP120 and started immunizing mice. And they realized very soon they realized that it is only giving non-neutralizing antibodies, not neutralizing antibodies. Because all the antibodies that are binding to GP120 is not binding or non-neutralizing when the trimeric quaternary conformation is present.
Okay.
And that became a challenge to maintain the quaternary conformation. And inactivated virus we cannot use.
Okay. Okay. I guess any question from all
Yes,
our speakers who are joining through Zoom? I think so. Dr. Nishira Sharma is also there. I think so. He's from the virus field. So if he has any question, Dr. Sharma, please.
Yeah. Dr. Shabir, nice talk. I mean, so could you please, show some light on the method you used to, specifically remove glycans from the virus surface?
Sir, can you switch on your video, please?
Okay. So these are all the glycosylation sites on HIV envelope are N-glycosylation sites. So they have a motif NXT, S or T. So N is the glycosylation site, any amino acid followed by serine or threonine. So we basically, because a lot of research has already been done and all the glycosylation sites had been identified. And so we knew that in the trimeric conformation from the crystal structure where those glycan sites are, and we specifically mutated those residues from N to Q. So there is no glycosylation site.
Okay.
So I think so. Dr. Sharma will also be joining for tomorrow's session. I think so. He has a talk later in the day as far as the session 1 is concerned. So thank you, Dr. Sharma, for joining. And any more questions from?
Yes, Ron, there is. So Dr. Shubhit, very nice talk.
Thank you.
And thank you for delivering such a nice talk. So we were whole day in the bacterial system. Now we came back to the viral system. So we had a change. It was a nice talk. So we have indeed few questions from the participants.
Mm-hmm.
So there are some. Okay, it's Ashwini Kumar from IIT Delhi. The question is, can a virus-like particle, VLP, without any genome be used as a vaccine candidate for HIV?
Yes, and that effort is also underway. And there is actually benefit that has more benefit than using a soluble form, because when you are using a VLP, you can decorate the surface with more antigens, and then it can be have the opportunity to induce better immunity and eliciting better antibodies. Okay. So yes, that is quite possible, and a lot of effort is under that way.
Okay, so another question, Ashish Kumar from NIT Rourkela. So the question is, can a VLP with mutated non-structural protein be used to induce humoral response?
Not really, because non-structural proteins, when we are talking about the virus as a whole, and that is causing the infection and entering to the host cell, are not exposing those non-structural proteins. Only so when you get an antibody against those, they will not be able to neutralize the host and the pathogen interaction. The only way to stop that is to have an antibody that blocks that interaction, either working directly at the site or working indirectly on the site, so that it changing the conformation and it is not binding to the host receptor.
Okay.
So I have one more question, Dr. Shabir, from your first part of the talk. So it seems from your study that the transmembrane region doesn't seem to be very important for post-fusion conformation and the trimeric structure, helix bundle formation, all those.
No. Yes.
So, but now the recent reports are coming that to showing some role of transmembrane domain, even some specific residues are involved in those. To bring out those conformations. So, how do you comment on that?
Yeah, you are absolutely right. So there are Kennedy epitope. There is one epitope that is in the transmembrane region that is called Kennedy epitope, and they're identified by Dr. Kennedy. And there is a C-terminal domain. And there, in a case of HIV, it is identified that there is antibody against those regions, and they are kind of neutralizing. So absolutely, those are neutralizing that I just said in that previous answer. And neutralizing antibodies are those antibodies that has to stop the infection, whether working directly at the site or indirectly at the site. So those antibodies that are binding to the HR1, HR2 region or those epitopes, they are blocking the formation of the pre-fusion to post-fusion conformation. So even if the host receptor is binding to that, the post-fusion conformation is not coming up because of the antibodies blocking is there. And that is how it is preventing the cells from the entry. And here comes the antiviral peptides.
Yeah.
So, antiviral peptides has a very potential roles in such mechanisms. In antiviral peptides, you don't really need to target the only neutralizing epitopes. Any antiviral peptide that can change the conformation of the spike protein or in such a way that it prevents the binding to the host receptor, that will really work well.
So they do have a role.
Yeah, they do have a role.
So, were there any more questions?
Yes, Dr. Shabir, there is one more question. This is from Satyajit Mishra from NIT Rourkela only. So the question is, can a chimeric antibody having good thermodynamic profile and suitably accepted as a broadly neutralized antibody be a better candidate than a traditional HIV vaccine?
Yes, and so the thing is here, there are already known. The first part is that to design an immunogen or a vaccine candidate that can elicit a broadly neutralizing antibodies. So here we don't have much control what kind of antibody is it eliciting, okay, and what percent of protection is there. But if there are already known antibodies like a PGD145 or PGD151, those are broadly neutralizing antibodies. Lot of efforts are on the way to increase the thermodynamics while retaining their broadly neutralizing property, increasing the thermodynamics and increasing the shelf life and half-life so that they can be used as a passive therapy. Now here comes the challenge in HIV with the passive therapy. Because passive therapy in HIV is not going to marketable in maybe it's a very good science, but not marketable because there is already ART drugs are available. Making those antibodies and selling them to them, that cost will not be possible in presence of ART. And ART is really working well. But yes, working on these aspects, people have identified mutations to increase the thermodynamics, increasing the shelf life, but retaining the bodily neutralizing property.
Properties. Okay. Oh, great. Okay, I think we got one more question now.
I would like to add on here,
Yeah.
there is also efforts are going on to make bispecific antibodies so that if we can block two neutralizing epitopes at the same time, with to give better, broader specificity and
wider specificity, right?
Yeah.
Okay. Oh, good. So I think just now one question came from Selina Patel again, NIT Rourkela. The question is, why HIV or coronavirus vaccine is not feasible, but anti-rabies vaccine is working with which is also virus. Okay.
Exactly. So one of the slide I showed the presentation of the number of antigens present on the surface. So in case of HIV, the number of antigens present is very less compared to chikungunya and dengue. So even in the case of rabies, it is very densely packed. So then many number of antigens are available. Number two is that those antigens are not so metastable. They don't fall apart so easily, not exposing the non-neutralizing epitopes.
Oh, okay.
So they give better neutralization, elicit better antibodies, and so they give better protection in case of that. But HIV is not the case.
Okay. Okay. I think there is no more question from the participant side. So we thank you for giving a nice talk. Arun, you can
There are no
continue.
more. Yeah, there are no more questions. Then I take this opportunity to thank Shabir and
Thanks,
also
Rohan. Thanks,
all the speakers, along with the presenters, along with our colleagues and all the participants. I think so. In the morning we had almost like 550 views for the first session, and I think so it will also be close to 300, 400 for this session as well. So I think so. The YouTube streaming of our live sessions, it's really productive. All of our students, they have also given a very good feedback. And thanks, Shabir, again for giving such a wonderful talk. And hope that we'll catch up soon. Although I can come to IISc anytime, but only the thing is because of this pandemic, I cannot come right now. So thank you very much, and thanks to all the participants and everyone out here. And we will start on time tomorrow. And again, this session is starting by 10 o'clock. And hopefully by 5 minutes before 10, we will be online. Thank you very much. And goodbye. Good night.
Goodbye. Yeah. Goodbye. Thank you.
Yeah, goodbye. And thanks. Thank you, everyone. Yeah.
Yeah.